Hey everybody, welcome back to the channel. Today I want to go through the entire biology section for the MCAT. I used these Milesdown Review Sheets and they were really helpful for me to be able to study for the MCAT. I got 100 percentile in the MCAT and I genuinely believe that everything that I needed to know for the exam were in these review sheets, and then all the practice exams helped me solidify the information. And so kind of want to help you speed run through these sheets and be able to go through them in an efficient way, and clarify any things that might not make sense. And so that's what I'm going to do in this video. So the first thing is parts of the cell. The nucleoid is the DNA region in a prokaryote. And it's really important to know that prokaryotes don't have a nucleus. They have nucleoids, right? And nucleoids are kind of this region where most of their DNA can be found, okay? The next thing is nucleolus. And these are in eukaryotes. And these are within the nucleus, but they don't have a membrane. And these are responsible for making ribosomes. And if you remember, let's go to nucleolus. You remember so this is kind of the way that, let's look at it better. So this is kind of the nucleus, and within it is a nucleoid, which is kind of this really dense material within the nucleus, right? This is the nucleoid over here. Yeah. And it's important to remember that they make ribosomes. And ribosomes, if you remember, are those things that are really important for connecting the amino acids so you can form the primary chain of amino acids that makes the eventual protein. It's important to know also that both prokaryotes and eukaryotes have ribosomes, because it's really important to be able to make proteins to do all the cellular functions that they need to do. Peroxisomes are things that break down materials, maybe like waste products or dangerous materials. Rough ER is responsible for accepting mRNA to make proteins, like we just mentioned. They're rough because they're studded with these ribosomes. Smooth ER are responsible for detoxing and making lipids. The Golgi apparatus is responsible for modifying and distributing proteins, and they're only in eukaryotes. And they're kind of like the packaging system of the cell. When I was studying this didn't make too much sense to me, the vesicular transport and cisternal maturation, until I finished my first year of med school. But I think looking at a picture would help. So cop to there. If you kind of look here, right? Or maybe let's try to find a better picture. This is a better picture. So here you can kind of very clearly see that, the side of the Golgi that faces the nucleus is called the cis side, and the side that faces the plasma membrane is called the trans side. And vesicles that go from the Golgi back to the ER are studded with these COP1, right, on their vesicle, and vesicles that go from the ER to the Golgi and fuse with the Golgi on the cis side are studied with COP2. Okay, and then anything that goes from the plasma membrane back to the Golgi or vice versa is studied with clathrin. Okay, so COP2 goes forward, COP1 goes reverse. That's anterograde is forward and retrograde is reverse, and clathrin is responsible for going from plasma membrane to Golgi and Golgi to plasma membrane. Okay, let's look. Peroxisomes are responsible for collecting and breaking down materials. Then gets repeated twice. And centrioles are 9 groups of microtubules, which are, as we'll see, parts of the cytoskeleton. And there are 9 groups of these microtubules, and 2 centrioles at right angles. And I can show you a picture of this. Centriole, centrosome. So you can see that. Let's see, what is this? Okay, so you can see that. Sorry. Okay, so you can see that there's 2 centrioles in right angles that form a centrosome. And this is a centrosome, these 2 centrioles, and they pull apart these chromosomes during anaphase, the centrosomes. Okay. Now we have lysosomes. Lysosomes are the recycling center. They're made by the Golgi and single membrane. And plasmids are within prokaryotes, and they carry DNA that is not necessary for survival. If you kind of remember, plasmids are those circular pieces of DNA that can replicate really quickly, and a lot of biochemists really like the plasmid because they can insert genes of interest to kind of farm genes of interest that they want. Okay, bacteria, you should know, can be in 3 shapes that are important to us. One is bacilli, which are rods. One is cocci, which is a sphere, and one is spirula, which is spiral. I kind of think that they look like pasta, which is how I remember it. Obligate aerobes are requiring O2. anaerobes will die in O2. They need to be in a non-oxygen environment. Facultative anaerobes can go between the two, and aerotolerant anaerobes don't use oxygen, but they won't die in oxygen. It's very important to know that when you gram stain a bacteria, it can either be purple or pink. If it's purple, it's a thick peptidoglycan cell wall. And if it's pink, then it's thin peptidoglycan cell wall, but it also has an outer membrane, which is outside that cell wall. Okay, now we have eukaryotes and prokaryotes. This is also very, very high yield. Eukaryotes have a ETC in the mitochondria, right? That ETC is called electron transport chain. They have large ribosomes, which these only have small ribosomes, and they reproduce via mitosis. Prokaryotes, on the other hand, have an electron transport chain in the cell membrane. They have small ribosomes. They reproduce via binary fission, which is a very, very similar process to mitosis. And plasmids carry DNA material, and they can have virulence factors, and plasmids can integrate into genomes, and when they're integrated, they're called episomes. Okay, some miscellaneous things that are not very high yield. Prions are infectious proteins. If you've ever heard of mad cow's disease or some really, really bad brain diseases, those are caused by prions. They trigger misfolding, and yeah, they're pretty bad. Viroids are plant pathogens. Okay, I kind of hinted at the cytoskeleton, but it's kind of important to know that microfilaments, again, pretty high yield. Microfilaments, actin is a very important one. Microtubules, tubulin is a very important one. And intermediate filaments, keratin and desmin. Those are important. Keratin and desmin is really prominent in your nails and hair. Microfilaments are the smallest, microtubules are the largest, and intermediate is in between those two. Okay. So intermediate filaments are very important for structural support. Microtubules are kind of what I mentioned, very important in the centriole and centrosome and moving things within the cell. And microfilaments are very important just in general and cell motility and things like that. Let's see, function. Microfilaments help generate forces used in cellular contraction and basic cell movements. Yeah, so they're very important in cell motility. Okay, tissues I was also very confused about until I finished my first year at med school. This is also kind of important. Epithelium is let's say any type of cell that covers the surface of some sort of body organ, right? Let's see if there's a better definition for that. Epithelium. Epithelia is a thin tissue forming the outer layer of a body surface. Or hollow structure. So anything that's covering the outer surface, or the body surface, or some organ or anything, that's an epithelium. Epithelium, there's several types. There's the parenchyma, which is the functional part of the organ. And then you can look at simple epithelium, that's one layer of cells. Stratified is when you have multiple layers of cells. Pseudostratified is really difficult to differentiate with stratified when you're just looking at it, like at a pathology slide. But pseudostratified, they're actually all one layer, but they kind of are different sizes, so they look like multiple different layers. Cuboidal is cube-shaped, columnar is, column-shaped, and squamous is very flat. Connective tissue is, anything, usually we think of that in the extracellular matrix, but there's something that supports, protects, and gives structure to other tissues and organs, right? And if I looked at an image, you can kind of see that they're like, you can kind of think of them as providing structure and providing support to organs. Connective tissue can be stroma, which provides support in extracellular matrix, and they're very important in bone, cartilage, tendon, blood. If you think of anything that supports and gives structure to something, those are bone and cartilage, tendon, blood. Those are things that are involved in connective tissue. Okay, next we have genetic recombination. This is how bacteria get new DNA. So there's transformation, which means that you're getting genetic information from the environment. There's conjugation, which means that that there's a transfer of genetic information via this bridge called the conjugation bridge. I kind of think of this like bacteria sex, even though I should probably not. But conjugation is kind of like how bacteria exchange DNA with one another. So if one bacteria is resistant to some sort of antibiotic, for example, it can pass that on through conjugation to another bacteria that is not. Transduction is when you transfer DNA using this virus called a bacteriophage. which are bacteria, viruses, and transposons are genetic information that can insert and remove themselves. And transposons are kind of these like sequences of DNA that can like jump around. Viruses. So I really enjoyed studying about viruses in my undergrad just because these things look really cool. Caspid is a protein coat. Envelope is just something that some viruses have. Virions are individual virus particles. So if you're talking about several virions, that's several virus particles, right? Bacteriophages are viruses that are specific to bacteria. And if you remember, transduction is how you transfer DNA between bacteria using these bacteriophages. Viral DNA. So viruses can have DNA or RNA. It could be single-stranded or double-stranded. If they're single-stranded, they can be either positive sense or negative sense, right? Positive sense is when you can take that DNA or RNA and you can take that RNA and you can directly translate it in the host cell. But if you have negative sense, then you have to use an RNA replicase and you have to convert it to the complementary strand, and then you can translate it. So it's one extra step within negative sense. Retroviruses are single-stranded RNA, and then a reverse transcriptase is first needed to make it into a DNA, and then you can make it again into RNA and then proteins. Bacteriophages can either be lytic or lysogenic. Lytic is when the virions are made until the cell just explodes. But lysogenic, the virus is basically dormant, and when stress activates that virus, then it goes into its lytic phase. All right, that's everything you need to know for the first page of biology. Okay, this is the second part of biology, which is reproduction. We're going to start with cell cycle. So the cell cycle is pretty easy to understand. First thing you need to understand is almost everything in the cell cycle is interphase except for this little bit, which is mitosis. And mitosis is the process of cell division where one parent cell makes two daughter cells. G1 is when the cell is making mRNA and proteins to prep for mitosis, right? G1 is this part. S phase is when the DNA is being replicated, right here. And the G2 phase is when the cell growth is happening. And so this is where there's organelles that are being formed and just overall preparation for mitosis. And mitosis, the M is mitosis and cytokinesis, which is where the DNA is dividing and into two daughter cells, and all the organelles are dividing into two daughter cells. Okay. So it's also important to know that during G1, the cell cycle can leave the cell can leave the cell cycle into G0, which is that if it doesn't need to divide, then there's no need to keep staying within the cell cycle and keep producing these two daughter cells. So it'll be in G0, which is kind of this dormant phase. There are growth signals, and I think this was very confusing to me. I pulled up a picture when I was studying, which helped me a lot. And so the thing to understand here is that let's use the color. Okay. When you're having cell division, some very important steps to just remember is that CDK and cyclin bind together to create a complex. That complex, CDK cyclin, phosphorylates RB to RBP, and then this changes shape and releases E2F. And when E2F is released, that causes a whole bunch of things that causes cell division to continue. And if this doesn't happen, if CDK and cyclin don't create a complex, then that's going to block the phosphorylation. That means E2F is attached, and that means that there's not that whole positive growth signal to cause the cell cycle to continue, right? And so if you look at maybe over here, you can see that CDK and cyclin can form a complex that causes, let me use my marker, that causes this RB to be phosphorylated. You have this E2F that's released, and that E2F causes the cell cycle to progress, right? And if this doesn't happen, then we're going to go this way, which means this E2F is bound and there's cell cycle arrest. Okay? So next thing is sex chromosomes, and this is pretty easy. Sex chromosomes, you should know that XX is females and XY is males in human beings. There are 23 pairs of chromosomes, which means that there's 46 total chromosomes. Out of those 46, one should be from your father and one should be from your mother, right? For each pair of those chromosomes. So 23 are from your mom, 23 are from your dad. X-linked disorders are when males express them and females can be carriers. And that's because if you have an X-linked disorder, since a male only has one X, and that X is from the mom, right? And the Y is from the dad. So if the male has an X-linked disorder, they're going to have the disease because they only have one X. Whereas females, they have two X's, one from their dad and one from their mom. So hopefully one of them is normal and the other one will contain the X-linked disorder. So they're just going to be a carrier, but they might not actually express the disease. A Y chromosome, the Y chromosome has very little genetic information. There's very few genes that result in some sort of phenotype. So there's very few. And a funny way you can remember this is sorry gene, which is sorry you're a male. Okay. The next thing I want to cover is mitosis and meiosis. I've kind of talked about mitosis a little bit, but I think it's helpful to just go through it really quickly. Okay, so the first thing to know is that in mitosis, I guess the first thing to know is that there's this thing called PMAT, which can help you remember it, which is that the phases are prophase, metaphase, anaphase, telophase. You might have learned about phases in between these. These are kind of the important ones for the MCAT. And that the ploidy is 2N throughout, and I'm going to talk about that a little bit more because that's important. But during prophase, the DNA is condensing, and centrioles are migrating to opposite poles. And when these centrioles migrate to opposite poles, these microtubules are forming. And if you remember, microtubules form these centrioles, and two of these centrioles form a centrosome. And we have the nuclear envelope disappearing. And basically what that's allowing is the DNA to be exposed so that we can bind the DNA with these microtubules and eventually split apart that DNA, because the DNA is in a condensed form called chromatin, heterochromatin. Okay. Next we have metaphase, and metaphase is when all the chromosomes are in the middle, right? Chromosomes meet in the middle, and that allows them to be now taken apart by these microtubules. Okay, the next phase is anaphase, and this is when we're pulling them apart. And it's important to know that now we're pulling each chromosome in half, right? Because first we duplicated these chromosomes, then we attached these chromosomes, and now we're pulling them apart. The next thing is telophase, and telophase is when these chromosomes are decondensing, nuclear membranes are reforming, and cytokinesis occurs. So this is basically the phase in which we're actually finalizing the process of making them into daughter cells. And I'm just going to show you a diagram so that this is all solidified in your head. So this is the diagram that I pulled up, and you can see that we have this spindle fiber, right? We have these centrosomes. We have these sister chromatids that are bound together, right? And we have, so this is interphase where everything is normal. Now these chromosomes start to condense, they've duplicated, they've attached at the center point, the spindle fiber has formed, and these centrioles are forming, and they have the spindle fiber. Now during metaphase, these all line up in the middle. You can see that the nuclear envelope has disappeared. Here it is disappearing, but here it's fully disappeared. And we've attached each of these spindle fibers to these sister chromatids. Now we're going to pull apart these sister chromatids, right? Okay. And then the last thing is we're going to form telophase. Telophase is where this nuclear envelope is reforming. We can see that it's still one cell technically. Cytokinesis has not fully occurred, but now cytokinesis is going to occur, and we can see that there's two daughter cells, and this looks just like how it was in interphase, right? So we can say that the ploidy is 2N throughout. 2N means that the number of chromosomes is always 4 throughout all of this, right? Even when we're in prophase, right, and even when we're in the even when we're divided into two daughter cells, we have the same amount of chromosomes. Now here, the chromosomes have doubled the DNA count, but it's attached, these sister chromatids are attached. So we're still calling it one chromosome, even though there's two sister chromatids on this. Even though here it's half the amount of DNA material, but this is just duplicated sister chromatids that are attached, and so we still call that one chromosome. That took me a long time to understand, so just make sure you understand that before moving on. The next thing is meiosis. So meiosis, I'm going to talk about this afterwards, non-disjunction, but that's a pretty important concept. So meiosis is pretty similar. We have prophase 1, which is where the chromosomes are again condensing. We have homologous chromosomes. And homologous chromosomes are basically one chromosome from your mom and one chromosome from your dad. So let's say we're talking about chromosome 2, right? You have two chromosome 2s, one from your mom, one from your dad. And here, instead of a single chromosome, which has sister chromatids like lining up in the middle at metaphase, we have the two homologous chromosomes are lining up in the middle, right? And we have a little thing called crossing over. So let me try to see if I can find a diagram for you. Prophase 1, meiosis, crossing over. So here you can see that, yeah, I think this is a good diagram. So here you can see that, normally when you have mitosis, you just have one of these that lines up in the middle. So let me use my marker. So normally in mitosis, in metaphase of mitosis, you have this thing that's lining up in the middle, and we're going to pull apart this chromosome using the spindle fibers from the centrosome, right? But here in meiosis, in metaphase 1 of meiosis, we have these two homologous chromosomes, which look very similar because one is from your dad and one is from your mom. And these two cross over, they exchange a little bit of genetic material here, and that basically contributes to genetic diversity. And what the purpose of meiosis 1 is to split apart these two chromosomes, right? We're not going to split apart this chromosome like we do in mitosis, right? We're going to split apart these two chromosomes, okay? And that's basically the purpose of meiosis 1. So if you understand that, then you're ahead of the curve, because that's a very difficult concept to understand. The next thing is anaphase 1, which is where we're splitting those apart. And then finally we have telophase 1, which is exactly what telophase was here, where the chromosomes are decondensing, nuclear membrane is reforming, sometimes it doesn't, cells are dividing, cytokinesis, and you have two haploid daughter cells of unequal sizes. The haploid daughter cells means that there's half the number of chromosomes that there used to be. And the reason for that is because we're literally taking two chromosomes and we're putting one in one cell and another in another cell. So obviously the two daughter cells of meiosis 1 is going to be haploid, right? Okay, now we got to the easy part, which is meiosis 2. And meiosis 2 is so similar to mitosis. Some people say it's like almost identical, which is very, very good for us because it's very easy to understand now. So now that we have these chromosomes in, let's see, meiosis 1 and 2. Okay, so now that we have, so we've split apart these chromosomes, right, in meiosis 1, and now we're here. And so now we have to go through prophase 2, which is where we have these haploid cells. And we're going to go through the whole same process of mitosis, which is we're going to line these chromosomes in the middle. And now we're going to split these chromosomes in half, right, by splitting apart the sister chromatids. Okay? And then, because of that, we're going to have telophase 2, chromosomes are decondensing, nuclear membrane is reforming, cells are dividing, and you have 4 haploid daughter cells. So we went from one parent cell, to 4 haploid daughter cells. The parent cell was diploid, the daughter cell is haploid. Mitosis, the parent cell was diploid, and the daughter cells are diploid, right? Mitosis is essentially, we're making the exact same daughter cells as the parent cell. Meiosis is we're creating some genetic diversity. We're trying to create daughter cells that look different and are actually genetically different because they're haploid. And there's a couple of things that could go wrong here, right? First thing is non-disjunction. And so if you have non-disjunction, this can happen during anaphase. And if this happens, then basically what you have during non-disjunction is these cells over here, sorry, these chromosomes over here are not splitting in half, right? And instead, one of these takes both. So let's just say that this centrosome here takes this whole chromosome and it doesn't split in half. And so what we're going to result with is a daughter cell that has an extra chromosome and a daughter cell that has one fewer chromosome. So that's pretty dangerous, and that's how we get things like Down syndrome, like trisomy 21, because there's a non-disjunction that happens and one chromosome doesn't divide in half, or that the homologous pair doesn't divide in half, and so we get aneuploidy. Aneuploidy is when we have a chromosome count that is not expected, right? Okay, and law of segregation is when we're having disjunction, so normal segregation. Okay, we made it through a huge topic, so just make sure you reread this and make sure you understand it. I should mention that P53 is a very important part of the cell cycle that's kind of responsible for a cell checkpoint. But yeah, we made it through a very important topic. So make sure you understand mitosis and meiosis really well, and you stare at those diagrams until it makes sense, because next we're going to go on to the male and female reproductive system. All right. So the next thing to consider is the semen. The semen contains sperm as well as seminal fluid, and seminal fluid is basically, I pulled up a slide, but seminal fluid is essentially anything that helps the sperm move. So fluid from the prostate and other sex glands that help transport the sperm out of the body. So they're responsible for transportation. Now the bulbourethral glands are something that helps make the liquid a little bit more viscous, and it helps clean out the urethra before the sperm get there. So if you want to see a picture, I hope YouTube doesn't take me down for this. But essentially the bulbourethral gland is this gland that helps, let's see, that helps produce a pre-ejaculate that cleanses and lubricates the urethra before the semen arrives. The next thing is the seminal vesicles and the prostate gland. So the seminal vesicles and the prostate glands, so the female reproductive tract is very acidic, and so the prostate gland basically produces a fluid that's very alkaline and helps the sperm survive in that environment. Now it's a little bit easier if you remember this pathway to understand the sperms. So it's 7 up, but the N is silent, so it's just SEVE up. And the S is for seminiferous tubules. This is where spermatogenesis, or the formation of sperm cells, first happens. Sertoli is responsible for spermatogenesis. Sertoli cells are somatic cells, so they're diploid, and they're essential for testes formation as well as spermatogenesis. Epididymis is something that stores sperm, and sperm are formed in the seminiferous tubules, and they're stored in the epididymis, and they gain motility here. The next thing is the vas deferens, and the vas deferens is the V here, and it raises and lowers the testes. So if you look here, it's kind of the thing that carries the sperm out of the testes. The next thing is the ejaculatory duct. And the ejaculatory duct is essentially just anything that the duct that delivers the sperm to the urethra. And the urethra is the part of the duct in which the urine or the urine or the semen is conducted outside the body. The penis is the main sexual organ that makes this all possible, which is used for insemination of a female, or as well as just the conduit for urine expulsion. So that is the male reproductive system. Just make sure you understand this. It's not super high yield for the MCAT, fortunately. Neither is the female reproductive system, but it's still important to understand all of this. All right, so the next thing we have to talk about is the female reproductive system. The first thing we want to talk about is ovaries. Ovaries have follicles that produce ova, and they're controlled by FSH and LH. And we'll talk about FSH and LH here, which are gonadotropin releasing hormones. But yeah, so I want to show you an image real quick, which is, so this is the ovary, and this is where oogenesis happens, right? And we're producing eggs in the female reproductive tract. Okay, so we produce the female gametes using oogenesis, and the ovaries is where we're producing the ova. Estrogen is responsible for developing the reproductive tract, and it thickens the uterine wall. And progesterone responds to LH, and it maintains and protects the endometrium. So how you can think of this is, so the endometrium is kind of this mucus lining that, so it's the mucus membrane that lines the uterus. And so when we have sexual reproduction, the male's penis enters the vagina, and we have the seminal fluid that's entering here, right? The sperm cells, and we want this uterus to be protected. And so what's happening is that the endometrium kind of thickens, and it's protecting the female reproductive tract in the uterus specifically. And essentially, the sperm is going to travel, and it's going to meet in the fallopian tube with ova. Okay, and so how I remember this is estrogen establishes and progesterone protects. And that's kind of how you can think of the endometrium and what estrogen and progesterone do. This is the pathway, so the egg, and then the peritoneal sac, and then we go to the fallopian tube. So the next thing is the gonadotropin releasing hormones, and we have FSH and LH. FSH is the follicle stimulating hormone, and you can kind of think of, the follicle. So it triggers spermatogenesis in males, And remember, Sertoli cells are kind of the thing that's responsible for spermatogenesis, so it's stimulating Sertoli cells. And the next thing is, in females, it's stimulating the development of the ovarian follicles, right, which are contained in the ovaries. The next thing is the LH, which is luteinizing hormone. And in males, that causes interstitial cells to make testosterone, which is kind of like the primary sex hormone for males. And in females, it induces ovulation. So it induces that that the gamete for the female to start moving around, the egg, right, in the female to start leaving the ovary and go through the fallopian tube. So if you want to look at this diagram real quick, the follicular phase starts with the first day of the period, and the luteal phase ends with the first day of the period, right? And this is kind of this 28-day cycle. And the FSH is about 13 to 14 days, and same thing with the luteal phase. And the FSH is kind of responsible for, remember here, it's triggering spermatogenesis, but in females, it's stimulating the development of the ovarian follicles. And the luteal phase marks the beginning of this ovulation period, where the egg is moving through a peritoneal sac in the fallopian tube. And we can see that estrogen is increasing throughout the follicular phase, and progesterone is increasing throughout the luteal phase primarily, but you can see that it spikes at the end of the follicular phase and in the first few days of the luteal phase, respectively. And so this can kind of tell you what the ovarian cycle looks like, how the FSH and LH respond at the end of the follicular phase and the luteal phase. And you should just know that, you know, the luteal phase is basically preparing the female for pregnancy, because, you know, you have increased levels of FSH and LH, and you have increased levels of estrogen and progesterone. And remember, estrogen and progesterone are responsible for maintaining and protecting the endometrium, right? So in case that the sperm does enter, we want to protect the endometrium. So that's what estrogen and progesterone are doing in the luteal phase. Okay, next section. Okay, the next thing we want to talk about is embryogenesis and development. And I'll be honest with you, I took an anatomy and development class in which the first unit we took was embryology in my undergrad years. And then we also studied this in med school, and I still found it very confusing to understand fertilization, morula, blastula, gastrulation, etc. Fortunately, you don't need to know it in a lot of depth for the MCAT. It is a pretty important topic, but you don't need to know it in too much depth. So knowing the basics really puts you ahead for this exam. The first thing is fertilization. So remember, the fertilization happens in the ampulla of the fallopian tube, right? The sperm has these acrosomal enzymes, and that's responsible for kind of penetrating the corona radiate and the zona pellucida. And it's very important that it has these acrosomal enzymes so that the sperm is able to enter, right, the female gamete. Then the acrosomal enzymes inject into the pronucleus, and we have cortical reaction that releases calcium, which depolarizes the ovum membrane and makes it impenetrable, right? We don't want more sperm to be entering in after the first sperm has already entered. So that's kind of the process, is the release of calcium, which depolarizes the membrane and makes it impenetrable after that. The next thing is morula, and so this is when we have a single cell, right? And we have a single cell, which is the embryo, and we want the morula. And so morula is kind of this solid ball of cells, and you can see that it's just a couple of cells, right? Like so it's just a solid mass of couple of cells, and it's very, very early on. Next, we have the blastula, and the blastula, I'll show you that too. Blastula is when we have a hollow, we start to get this blastocele in the blastula, right? And so the cells, you can see that there's this inner cell mass, right, which is right here. There's this inner cell mass, and then there's this blastocele, which is this hollow space. And then we have this trophoblast, which is the outer cell membrane. Not cell membrane, but just outer, I guess you could call it an outer membrane, right? And that's called the blastula. So we're starting to get a little bit more of polarity established in this, right? We see that there's a fluid-filled blastocele. We have implants it's implanted into the endometrial lining. So if you remember from the past couple of minutes, the endometrial lining is where we finally implanted the blastula. And then we have the trophoblast, which eventually will become the chorion or the placenta. The trophoblast is that outer thing I was showing you. I'll show you again. So remember, that this thing is the trophoblast over here, right? This outer lining of cells, and that's eventually going to become the placenta and the chorion. And then the next thing is the inner cell mass, which is going to become the organism. So that's what, this thing over here is the inner cell mass. That's going to become the embryo, the organism. Okay. So gastrulation is this process in which we're forming the ectoderm, mesoderm, and endoderm. And this is very important and high yield for the MCAT. They're often going to ask you like, oh, I don't know, the mouth. What layer did that originate from? And you want to know that that's from the ectoderm. Fortunately, this took me a long time to understand. But fortunately, there's just this really easy mnemonic that you can just, or not mnemonic, but just a way to remember it. Ectoderm is kind of like the tractoderm. It's your brains, your nervous system, your skin, your hair, your nails, your mouth, your anus. Everything that people are attracted to, your nervous system. So like your smarts, your skin, your hair, your nails, your mouth, your anus, everything that people are attracted to, that's your ectoderm. That's a very easy way to remember it. Mesoderm is your musculoskeletal, your circulatory system, gonads, adrenal cortex. Things that how I remember this is kind of like the things that are responsible for movement, like the musculoskeletal system, the circulatory system, so like all your blood and everything, your gonads, your adrenal cortex, which makes those steroids, right? Those are all kind of movoderm things. That's how I remember it. Endoderm are things that are inside. So the endocrine glands, the GI tract, the respiratory tract, the bronchi, the bladder, the stomach, all these things are endoderm, right? If these ways of remembering it don't work, just make sure that you remember these things because it's very important that you remember it. Okay, the next thing is neurulation. Neurulation is when you've developed a notochord. The mesoderm forms that notochord, and the notochord induces the ectoderm. The ectoderm forms neural folds and neural tubes, and the neural folds form the peripheral nervous system, right? The neural folds will become neural crest cells, which form the peripheral nervous system, and the neural tube forms the central nervous system. And I really encourage you to watch a video, an animation video that shows neurulation in an embryo. It'll help you kind of understand three-dimensionally what's going on. I will say that this is a very, very confusing topic the more that you study it. And I know like physicians that are extremely brilliant that have taught us and just say like, I don't fully understand what's going on because it's just very complicated. But you should know that neurulation is this really important process that's establishing what's the peripheral and what's the central nervous system. And you should remember that the nervous system is part of the ectoderm. Okay, the next thing is stem cells. Stem cells can be totipotent, pluripotent, and multipotent. Totipotent, any stem cell can become any type of cell. Pluripotent is that it can become any type of cell except for the placental structures. And multipotent is that it can become multiple different types of cells, but not any type of cell, right? So you should know that adult stem cells within the adult body are multipotent, right? And usually you have to put in transcription factors for it to become the type of cell that you want. But if you take, for example, cells from, I'm guessing like the morula, for example, right? Then that might be a totipotent cell, which means that it can become any type of cell because it hasn't differentiated yet. Pluripotent, probably from the inner cell mass, but again, I'm not entirely sure. But you should just know that these are the degrees of stem cell. Stem cells are cells that can become multiple different types of cell types. Okay, fetal circulation. This is a pretty interesting topic. So you should know that the embryo doesn't really take oxygen from the air. It's kind of taking it from the mother, right? And so it takes it from this placenta, and we have an umbilical vein that goes to the embryo, an umbilical artery that goes away from the embryo, right? So just remember that the vein goes to the embryo, the artery goes away from the embryo, and these are called the umbilical artery and umbilical vein. And the fetal hemoglobin has to have greater oxygen affinity than the adult hemoglobin. And the reason for this is because the fetal hemoglobin, they need to like really, really want that oxygen in order to get it. And so that's why the oxygen diffuses from the placenta, from the mother to the embryo. And so this oxygen and carbon dioxide exchange happens through diffusion because the fetal hemoglobin wants that oxygen more than the adult hemoglobin. Okay, I'd say that these are kind of all things that you should just look at and kind of be familiar with. It's not too important that you have memorized every single thing, but it should just spend like a couple minutes and make sure you kind of understand this stuff. All right, twins can be either dizygotic, fraternal, or identical. I'm sure you know a twin, and you're like, either they're identical twins or they're fraternal twins. If they're identical twins, that means that they came from the same zygote, and fraternal means they're different zygotes, right? Cell specialization. Determination is when a cell determines to become a specific type of cell. And differentiation is after determination, where you're actually selectively transcribing the genes that result in the cell-specific function. So the reason why a cell from the lung is different from a cell from the stomach, for example, is because it's not because the genes are different. It's because we've selectively transcribed different genes in different cells so that it only expresses those specific types of genes. And as a result, we have a different type of cell. And so that process is called differentiation. Induction is when a group of cells influence the fate of nearby cells. This is mediated by inducers, which are also commonly called growth factors. I'm sure you've heard of them. We have signaling. Signaling can be autocrine, which is a cell signals to itself. Paracrine, which means a cell signals to a nearby cell. Juxtacrine, which means it goes to a nearby cell again through this junction. And endocrine, which means it goes through the bloodstream. And you can think of endocrine glands, and that means they're releasing hormones through the bloodstream. Finally, we have fetal shunts, and fetal shunts are very important because the embryo hasn't fully developed, and so you can't expect the embryo's lungs to be or the embryo's heart to be pumping completely, or the embryo's lungs to be providing oxygen, right? So the blood supply has to skip certain parts, and this took me a long time to understand. But if you just understand that, then you're good. So how it skips the lungs, because the lungs are essentially full of fluid. It's not actually containing oxygen. It's not breathing in or anything. So it has to skip the lungs, and that's through the foramen ovale. It has to skip the lungs, which is also through the ductus arteriosus. And the foramen ovale goes from the right atrium to the left atrium, and so it basically completely skips the lungs. And the ductus arteriosus goes from the pulmonary artery to the aorta, which again is skipping the lungs. So it's it's a right to left shunt. The liver, it skips the liver because the umbilical vein goes to the inferior vena cava. So it just completely skips the liver, and the umbilical vein, which is supplying oxygen from the placenta, goes to the inferior vena cava, which will go to the right atrium. Okay, very good. Next, we have the nervous system, which is pretty exciting stuff. Okay, so you should know that there are 3 different types of neurons. One is afferent, one is efferent, and one is interneuron. Afferent goes up the spinal cord to send the signal. Efferent exits the spinal cord to again send the signal to perhaps your musculoskeletal system. And interneurons are between those neurons. You can sum up these signals from neurons. So you can imagine that these nerve terminals are going to these dendrites, going to other axons. Sorry, let me show this real quick. So you can imagine that there's these nerve terminals of another neuron, and there's another one that's here, and there's another one that's here. And all of these are sending signals, right? Some of them might be sending signals that is like, oh, go, go, excitement. Some of these might be sending signals that are inhibitory, right? But all of these neurons that are surrounding this one neuron might be sending it signals, right? And so you can send signals temporally or spatially. You can sum up those signals. Temporally means that there's same space but different timing, and spatially means the different space but same time. Action potentials, basically you have a stimulus, and if the stimulus, if it exceeds the threshold, then we have this whole depolarization, repolarization, refractory period, and back to the resting state. But if it doesn't go to the threshold, then it's a failed initiation, and so we won't have any action potential, right? So the stimulus from other neurons, for example, has to exceed the threshold to cause depolarization. Depolarization is when the sodium channels are opening. And you should know, definitely know, that if you have a neuron, right, the sodium is very high on the outside and very, very low on the inside. And the potassium is very high on the inside and very low on the outside. And so if you remember that, that's a really easy way to remember that when the sodium channels is opening, the sodium is going into the neuron, right? And that causes it. We have a lot of positive ions that are going into the neuron, so this voltage goes up. And then we have repolarization, which means that once it gets to a certain positive voltage, these potassium channels start to open, right? And potassium is leaving the neuron, and so we have repolarization. It's going down because a lot of positive ions are leaving the neuron. And it goes a little bit more down than the resting state, right? And this is the refractory period. The refractory period is this period where it can't really receive another stimulus. And if it does receive another stimulus, it has to go really far way up to meet the threshold. So there's like an absolute and effective refractory period, which it basically means that the neuron is probably not going to be excitable during that state. And then it goes back to the resting state and it's ready to be stimulated again for another action potential. Okay. You should know that there's also a sodium-potassium pump, and the sodium-potassium pump essentially responsible because we have had this whole process of the sodium coming in and the potassium coming out. We need to make it go back to sodium being outside and potassium being inside. And so we use this ATPase. We use ATP to restore the sodium outside and the potassium inside. And so that's what this ATPase does. It sends sodium outside and sends potassium inside. Next we have this diagram over here, and this is a synapse. This is what I was trying to show you, which is that the dendrites of one neuron are receiving signals from the nerve terminals from the axon of another neuron, right? So this is the axon, this is the nerve terminal, and this is the dendrite, and this is the receptor. And this is a chemical signal that's being sent from the axon of one neuron to the dendrite of another neuron. And this chemical signal is called neurotransmitters. And the neurotransmitters are within these synaptic vesicles. And synaptic vesicles will be released into the receptors of the dendrite of the next neuron. And how that happens is that there's this action potential, which is sending this electrical signal down, 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 releases, and once this electrical signal goes down, the calcium channels open. Calcium is also high on the outside, so the calcium goes inside. This causes the synaptic vesicles to go to this docking protein, and basically exocytosis happens, and these neurotransmitters, which were inside the synaptic vesicles, go to the receptor of the dendrite of the next neuron. And this is called a postsynaptic cell, right? This is called the postsynaptic cell. This is called the presynaptic cell. And this space over here, this cleft, is called the synapse. Okay, so we can remove the neurotransmitter, and this is pretty high yield actually. We can remove these neurotransmitters in this synaptic cleft by breakdown of enzymes, reuptake, and diffusion out of cleft, right? And so the reason why this is kind of important is because if we want to sort of manipulate different things, then we can manipulate these things and we can cause neurotransmitters to be removed. And so if you think about things like SSRIs, which are made for people who have depression to have, for serotonin to have more impact, then that's kind of one of the ways that we manipulate this process for SSRIs to work. And so sometimes in the MCAT they'll give you a passage and they'll ask you about a drug and they'll ask you, does that cause the neurotransmitter to have lesser impact on the postsynaptic cell? And so that's kind of a way that you can think about it. Okay, so I kind of already mentioned this, but this is the dendrites. This is the cell body. It's also called the soma. This is the axon, right? This is the nucleus over here. So the nucleus is contained within the cell body. And these are the nerve terminals. The nerve terminal is what sends the neurotransmitters across the synapse, right? You should know that this thing is called a myelin sheath, right? And myelin sheath basically helps this conduction, so electrical conduction, go faster. That's called saltatory conduction. And these little spots in which there's no myelin are called nodes of Ranvier. And this is called saltatory conduction. And you should know that there's Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. Next, we have glial cells. Glial cells are kind of what I was just talking about Schwann cells and oligodendrocytes. And I want to give you a definition of glial cell. So glial cells are things that provide physical and chemical support to your neurons. So we have astrocytes. Astrocytes are responsible for that blood-brain barrier. It's very important that you have this so that, if you're giving a drug to somebody and maybe it has some cytotoxic effects, you don't want it to go to the brain. And so that kind of creates that if there's these chemicals in your blood, you don't want it to go directly to your brain. And so there's these astrocytes that are helping controlling solutes go from the bloodstream to the nervous tissue. Something that people ask is like, oh, does that mean the brain doesn't get blood? And no, brain definitely gets blood. It's just that there's this blood-brain barrier that makes sure that these solutes are not cytotoxic. The next thing is ependymal cells. I don't actually know how to pronounce it, but this creates a barrier between the cerebrospinal fluid and the interstitial fluid of the CNS. Next thing is microglia, which digest waste in the CNS. And then, like I just said, the Schwann cells and the oligodendrocytes make myelin, Schwann cells in the PNS, and oligodendrocytes in the CNS. I will say that it's not super high yield for the glial cells is not a very high yield topic, but action potential, synapse, these summations, neurons, and this process is a very high yield thing. Okay, next thing is white gray matter and gray matter. White matter is myelinated sheets, and gray matter is the cell bodies and dendrites, which are unmyelinated. It's important to know that in the brain, the white is deep and the gray is outer, and in the spinal cord, it's reversed, where the gray is deep and the white is outer. So I always kind of make this diagram for myself, which is this is the brain, and I just want to remember that outside is gray and inside is white, right? And in the spinal cord, I just make the same diagram for myself, and I just remember that the inside is gray and the outside is white, right? So just remember that they're reverse. The next thing is a reflex arc. Reflex arcs are kind of like when the doctor taps your knee and you want it to kind of jerk without you doing anything. And that can be monosynaptic or polysynaptic. It can have a sensory neuron and then a motor neuron. So the sensory neuron can receive the signal, and the motor neuron immediately responds without any interneuron. And polysynaptic is when you have an interneuron in between. Central nervous system is the brain and spinal cord, and the peripheral nervous system is everything else that's a neuron in the body. The peripheral nervous system, and this is very high yield for you to know, can be somatic or autonomic. Somatic means voluntary. Autonomic means that it happens without you thinking about it. And some things in your body, you just want it to be autonomic, right? Examples are when you're digesting food, you're not thinking about the acid that's being released in your stomach and, the intrinsic factor that's being released by your parietal cells and all those things, right? Same thing, like when you're breathing in, you're not thinking about all the bronchi and everything. So you want some things to be autonomic. You also want some things that be somatic. And so somatic, sensory means afferent, and motor means efferent. So if you sense something that's a afferent neuron that's feeling it, sending it to your brain and spinal cord, and the motor is the efferent, which is the response, right? Autonomic, it can be either sympathetic or parasympathetic. Sympathetic means it's a fight or flight response, and parasympathetic means it's a rest and digest process. So how I think about it is if you're in a campfire and you're seeing a bear, that's fight or flight, right? But if you're just eating food by the campfire, that's rest and digest. And so it's pretty easy to remember what happens in the sympathetic if you kind of just understand that concept, which is that in fight or flight, we have your bronchi are relaxing, your blood is moving to locomotion. So like, for example, if you need to run, so your blood is moving toward that. Peristalsis is decreasing. Peristalsis is the movement of your food through your digestive tract. The neurotransmitters are acetylcholine for preganglionic and epinephrine and norepinephrine for postganglionic. Rest and digest, you have reduced bronchi, right? Because you have conservative energy, increased peristalsis because remember you're digesting, and your preganglionic is acetylcholine, and your postganglionic is also acetylcholine. So it's only and you can remember this because epinephrine and norepinephrine is just adrenaline basically. So if you hear somebody like, oh, my adrenaline spiked during that exam, or like when I was running from that bear, that my adrenaline spiked, that's because your epinephrine and norepinephrine are the postganglionic thing. So after the ganglion, the postganglion, that's what the neurotransmitter is. Okay, great. I hope that makes sense. Just make sure you review this because this is all very high yield information. The next concept is the endocrine system, which is also very, very high yield. All right, so this is the endocrine system, and I want to say that out of everything that we've looked at so far, this is probably the most high yield information that I'm going to tell you, and especially this part. If you kind of know these endocrine organs and hormones, then you're probably going to get a good number of questions right, just because this is a lot of the things that are tested in the biology section of the MCAT. So the first thing you want to know is that there's a difference between peptide hormones and steroid hormones. And the main thing is that peptide hormones, because they're made of amino acids, are soluble, and steroid hormones, which are made of cholesterol, usually are not soluble. So peptide hormones are cleaved from larger polypeptide. Golgi modifies and activates these hormones. They're put into vesicles and released by exocytosis, right? They leave the cell, and they are polar, which means they cannot pass through the membrane. So they use an extracellular receptor like GPCR, which is what we're going to talk about right here. And so because they're polar, and remember that the cell membrane is full of lipids, right? They cannot pass through that membrane, so they have to use receptors on the cell membrane and then cause a variety of effects inside. And so there's a lot of secondary messengers that are very common that you'll see in the MCAT, which is like CAMP, CA2+, IP3, right? And CAMP being probably one of the most common secondary messenger. Steroid hormones are nonpolar, and that means they can pass through the membrane. They're usually made in the gonads and the adrenal cortex, right? And they're made from cholesterol, and they can dissolve. They do not dissolve, so they have to be carried by proteins. So just remember that this can dissolve in water but can't move through the cell membrane, and this can't dissolve, so must be carried by proteins but can pass through the cell membrane. And they activate nuclear receptors. So instead of a receptor on the cell membrane, they activate receptors inside the nucleus, and that has direct action on the DNA. So examples are things that we've looked at already: estrogen and testosterone, as well as cortisol. Okay. Amino acid derivative hormones, there's not too much to say about this. They share traits from both peptides and steroid hormones. So catecholamines, for example, they use GPCR. Thyroxine binds intracellularly, not too high yield. GPCRs are pretty high yield. And so you should know that usually what happens is that epinephrine is a first ligand messenger. It binds to the G protein coupled receptor, which then activates the G protein, right? Which then causes adenylyl cyclase to convert ATP to CAMP. And remember, I said CAMP is one of the most common secondary messengers. And then CAMP causes protein kinase A to promote glycogen breakdown, right? So this is going to happen, the glycogen breakdown, and it's going to inhibit the synthesis of glycogen, right? So that means that we're going to have breakdown of glycogen, right? That's an example of what GPCRs do. And you should know that at the end of this process, phosphodiesterase deactivates the CAMP, and GTP is hydrolyzed back to GDP. And this makes sure that the next time that an epinephrine molecule binds to the GPCR, this whole process can happen again. Okay, we can have direct hormones and tropic hormones. Direct hormones act directly on the target tissue or organ. An example is insulin. And tropic hormones require an intermediary. So as we're going to see, GnRH and LH are both tropic hormones. Okay, you probably know that diabetes is when there's too much sugar in the blood. There's two types, two ways that this could happen essentially. One is that we don't produce enough insulin, and so glucose is not able to enter the cells. Another one is that the insulin receptors are desensitized, so they work less efficiently, and so glucose is still unable to enter the cell even if we have enough insulin, just because the receptors aren't working. Okay, so I want to say that I can help you through all of this, but I think most of it is going to come down to your memorization of it. I can walk you through some of the ways that you can memorize all this, and I will say that it does look really complicated in the beginning, but the good thing is that you probably already know a lot of this from your biology classes, even if you don't know it. Or just general information. For example, the adrenal cortex, you might know, makes cortisol, aldosterone, androgens. You might know that the pineal gland makes the melatonin, which is responsible for the sleep cycle. You might know, or we already know, that the gonads produce the testosterone, estrogen, and progesterone. We might know that pancreas produces insulin and glucagon, as well as somatostatin, which I'll talk about. But essentially, I want you to know that you shouldn't worry too much about this. It shouldn't take too long to memorize. The important thing is that you look at it every so often, and you focus on spaced repetition to make sure that you understand all of this. It is very, very high yield that you know this, and so that's a good thing, is that you're going to differentiate yourself if you know all this information. So really quickly, I'll run you through this. The hypothalamus has GnRH, GHRH, TRH, CRH, dopamine, ADH, and oxytocin, right? So basically, I'll talk about ADH and oxytocin. This is produced in the hypothalamus, but it's released from the posterior pituitary, right? So if you look, so this is ADH and oxytocin are produced in the hypothalamus, but they're released, as you can see, in the posterior pituitary. Dopamine decreases prolactin, which we'll also talk about. CRH increases ACTH, right? And ACTH is something that we'll talk about when we get to the adrenal cortex. TRH increases TSH, right? And TSH is something that we'll talk about in thyroid glands. GHRH and GnRH are pretty. So GnRH is responsible for increasing FSH and LH, which you can remember are things that are very important in the male and female reproductive cycle, especially in the female reproductive cycle. GRHRH is something responsible for increasing the growth hormone. Okay, so make sure you understand all this. The good thing is that a general theme of the hypothalamus is that it produces a lot of these hormones that are responsible for increasing the production of something else. So yeah, the one thing I misspoke about that I want to correct is that the TRH increases TSH, which is responsible in the anterior pituitary. I think I said thyroid gland, but that's actually something that's important in the anterior pituitary. Okay, the next thing is the pancreas, and as I mentioned, there's insulin and glucagon, somatostatin. So insulin is in the beta islet cells, and they decrease the blood sugar, right? So diabetes is when you have too much blood sugar. Your insulin is either not sufficient or the receptors are not working. Glucagon is in the alpha islets of the pancreas, and it's responsible for increasing blood glucose. Somatostatin is in the delta islets, and it's responsible for decreasing insulin and decreasing glucagon. Gonads are responsible for testosterone in testes and estrogen and progesterone in ovaries. Remember, estrogen establishes, progesterone protects. Testosterone is responsible for just the general male sexual characteristics. Pineal gland is responsible for producing melatonin, and if you ever heard of like melatonin sleep supplements, that's something that helps you sleep because you can kind of use that to remember that melatonin controls the sleep cycle. Next, we have the anterior pituitary, and this is kind of the most important one because it has so many important hormones. So the way that I remember this is flat peg. Flat peg is FLATPEG, right? F is for FSH, which again, remember, FSH is responsible for spermatogenesis and growth of these ovarian follicles, which are responsible for the gametogenesis in males and females. LH is responsible for the production of testosterone as well as inducing ovulation during the luteal phase of females, and you can scroll back if you want to remember that. The ACTH is responsible for synthesizing and releasing glucocorticoids from the adrenal cortex. So the ACTH, we'll see, is responsible for causing these things in the adrenal cortex. TSH, remember, TRH over here promotes the production of TSH in the anterior pituitary, which then synthesizes and releases these things, that triiodothyroid, I've never known how to say these things, and thyroxine from the thyroid. So you can see from here is going to cause these things. Prolactin increases milk production, endorphins decrease pain, and growth hormone increases growth of bone and muscle, and increases growth in bone and muscle. I don't know why I said twice. Thyroid gland is responsible for T4 and T3, and that's made by follicle cells, and it causes the basal metabolic rate to increase. And calcitonin is made by parafollicular cells, and they increase the calcium in bone, they decrease the calcium in blood, they decrease the calcium absorption in gut, and they increase the calcium excretion for kidneys. So calcitonin is a very important hormone when you have too much calcium in your body, in your bloodstream. Okay, the very good thing about parathyroid gland is that PTH basically does the opposite of calcitonin. It's a very easy way to remember it. It decreases calcium in blood, decreases calcium in bone, increases calcium in blood, increases calcium absorption in gut, decreases calcium excretion in kidneys, and then because of the bone breakdown that's happening here, we have more calcium in the blood, and it also activates vitamin D. And vitamin D is another way of saying calcitriol. Okay, remember I said that the hormones of the posterior pituitary are produced in the hypothalamus and released in the posterior pituitary. ADH is a very important hormone that comes up quite a bit in the MCAT, and it's responsible for decreasing the urine output in, decreasing the water output in your urine, and also causing vasoconstriction. And when your pee is really, really yellow and very concentrated, you can think that that's happening because these ADH are decreasing the H2O output, and so you have a lot of solutes that's in your urine. Oxytocin is responsible for a lot of this bonding behavior, increased production of milk, uterine contractions, has a positive feedback cycle. Okay, adrenal cortex, remember that the ACTH is synthesizing and releasing glucocorticoids from the adrenal cortex, and the adrenal cortex is releasing glucocorticoids like cortisol. You should know that cortisol is one of our stress hormones. It increases glucose, decreases protein synthesis, decreases our immune system, right? And it's kind of when you have a lot of stress, you increase your cortisol production. Mineralocorticoids like aldosterone decrease the potassium in the blood, increase the sodium in the blood, increases the water in the blood due to osmosis, and increases the blood pressure. So basically, when you have less blood pressure, your renal artery is sensing that and telling the adrenal cortex to release aldosterone. And that aldosterone will activate these channels in your collecting ducts, which tells those channels to release more potassium in your urine and reabsorb more sodium from your urine back into the bloodstream. And so that's what's happening here. And the water follows the sodium. And this is essentially a way to prevent your blood pressure from going low, and so it increases your blood pressure. Finally, we have androgens, which are converted to testosterone and estrogen in the gonads. Okay, lastly, we have the adrenal medulla, and this is kind of responsible for the catecholamines. Remember, like the adrenaline type of things, epinephrine and norepinephrine, and they do very similar things. Epinephrine is an antihistamine, and they both increase blood pressure and heart rate. Let's go. We finished the very high yield part, so now we're going into the respiratory system. Okay, next we're talking about the air pathway. And the air pathway is consisting of, the nostril. Then we have the pharynx. Pharynx is where food and air travel through. And then we have the larynx, which is where only air is passing through, right? You don't want the food to go through the larynx because then you're going to have some sort of obstruction in your larynx, right? And this contains your vocal cords. Next, we have the trachea, and this is consisted of ciliated epithelium, and it's collecting all the debris. And then we have the bronchi, and the bronchi is also collecting the debris, but it's important that this is the pathway through which air is passing through, right? Now we get to the bronchioles, which are the smallest branches of the bronchi. And finally, we get to these little things called alveoli, and they're these little sacs in the lung, right? So if you imagine, we have these branches all over, and we keep getting tinier and tinier until we just have these little branches that are connecting off the bronchiole, and these little sacs are called alveoli. And these sacs is where diffusion occurs. So you have all your blood that contains carbon dioxide coming from your pulmonary your pulmonary artery contains all your deoxygenated blood, and that's conducting diffusion with your alveoli. And because of that diffusion, the alveoli is giving oxygen, and your pulmonary artery is giving carbon dioxide. Surfactant is something that's responsible for reducing surface tension, and so surfactant is really important in preventing your sacs, your alveoli, from collapsing. And in med school, you'll learn about some diseases like neonatal respiratory distress syndrome, where there's not enough surfactant, and so these babies are born with too much surface tension, and so there's collapse of their alveoli. Okay. You should know that pulmonary arteries are going away from the heart and pulmonary veins are going toward the heart. How I remember that is arteries start with A and they always go away from the heart, right? The next thing is spirometry. Spirometry is something that's really common in the allergy clinic if you ever go to one. And that's kind of a way to measure your lung capacity and measure, when you're inspiring and expiring, how much air is going in and out. And it's this really cool machine where you just basically breathe as hard as you can into this machine, and it'll calculate how much air is going in. And so you can calculate all these different metrics. It doesn't calculate how much total volume is in your lung, but it does give you a lot of important metrics. So this is not a super clear diagram, so I want to give you a little bit better of diagram, volume diagram chart. Yeah, this is good. It might be too complicated. Let's use this one. Okay, so this is a diagram of your lung. So the tidal volume is how much air you're breathing in and out whenever you're like how I am right now, where I'm not like trying to breathe really in or really out. It's just my normal breathing volume. That's the tidal volume. This is usually like 500 milliliters or so. Then we have the inspiratory reserve volume, which is what I just did. When you try to inspire as much as possible, how much do you go up to, right? That's your inspiratory reserve volume. This, let me use my marker. So this is the inspiratory reserve volume. This is the residual volume. This is the expiratory reserve volume, which is that if you breathe out as much as you can, right? How much of that air can you breathe out? That's your expiratory reserve volume. And then finally, you should know that when you try to breathe out as much as possible, there's still this little bit of air within your lungs that you're not able to expel, and that's called the residual volume. Okay? Now we have capacity. So inspiratory capacity is the inspiratory reserve volume plus your tidal volume. Your functional reserve capacity is your expiratory reserve volume plus your residual volume. Your vital capacity is your expiratory reserve volume plus your tidal volume plus your inspiratory reserve volume. And then your total lung capacity is everything, right? And I wish I had a better way for you to memorize the capacities, but you just kind of have to memorize it. I forget it all the time in med school, so it's important to just it's not that hard to remember the volumes, but the capacities you just kind of have to memorize, and that's what those terms are. So now we have the medulla oblongata, and the medulla oblongata is kind of responsible for sensing when you have too much carbon dioxide or too little oxygen, and tells yourself to respirate more or less. And so it's really important to know that whenever you have too much carbon dioxide, build up in your body, your body is going to tell yourself that you need to breathe faster. And so you will have more respiration. You might breathe like 26 breaths a minute, for example, instead of like your normal 18 to 20. And so what you're trying to do is breathe out that carbon dioxide. And so when you have too little carbon dioxide, you're going to actually decrease the number of breaths you're taking because you don't want to do a lot of breaths and get rid of that carbon dioxide. So that's basically what the medulla oblongata is helping you do. And when you have too little oxygen, you're going to increase ventilation again. Okay. Whew. It's good to know what the pleura is. The pleura is basically this connective tissue that's surrounding your lungs, and the very outer layer of the outer pleura, called the parietal pleura, that's composed of one layer of epithelium, right? So the pleura are this thing that surrounds the lungs, and there's two layers of that pleura. There's the parietal pleura, which is the outside one, this red thing over here, and there's the visceral pleura, which is this blue thing over here, right? Okay, then you have a space in between, which is called the pleural space. And when you start med school, you learn about a couple of different diseases where there's like fluid buildup or different types of pressure that's in this pleural space that's causing your lungs to kind of collapse. And then you have your lungs, which are these huge sacs that surround your heart. And you can see that it's very easy for your heart to send blood out to these lungs, and then there'll be a passive diffusion between the alveoli and these arteries, the pulmonary artery from the heart. Okay, next we have inhalation. Inhalation is negative pressure breathing. It's an active process. The diaphragm and the external intercostal muscles are contracting. You have increased intrapleural space, increased thoracic cavity, and decreased pressure. This is kind of difficult to understand. So how I think of it is when I'm inspiring, right? When I'm inspiring, basically what's happening is I'm taking air inside, right? But how does that air want to come inside? Basically because of a pressure difference, right? How do I create that pressure difference in the first place for the air to want to come inside? Basically, I'm causing my diaphragm to kind of go down, right? And because the diaphragm is kind of going down, what's happening is that there's more volume in my thoracic cavity, right? And so what's happening is because there's more space in my thoracic cavity, there's more space in my intrapleural space, there's less pressure, there's more space, right? Less pressure. And so the air from outside is slightly higher pressure, and so it comes in. So that's basically how I'm doing it. I'm contracting my diaphragm and my external intercostal muscles. I'm creating more space in the intrapleural space in the thoracic cavity, and so I'm decreasing the pressure. Next, we have the lung volume and the lung pressure. So the lung volume increases, like I just said, and the lung pressure decreases, and so air is rushing in. Exhalation is the exact opposite process. The only thing is that inhalation is an active process where I'm contracting, and exhalation is kind of a passive process where all I'm doing is just relaxing my muscles. There's less volume in the lungs, and so there's greater pressure, and so the air is rushing out. And sometimes I have to do active exhalation where I have to use all of my muscles to really force myself to exhale the air, and that can happen in a couple of different diseases, or if I'm just really, really trying to get rid of the air. I will say that this is not too important for you to memorize in terms of high yield. So just kind of look over this. The mucous membranes are kind of important for protecting against pathogen, because when you're breathing in, you might inhale some pathogens, and so your mucous membranes in your in your lung parts, like your trachea, your bronchi, they kind of have this mucous membrane that protects against these pathogens. Lysozymes are in your nasal cavity, in your saliva, and they're attacking gram-positive peptidoglycans. So you don't want to breathe in gram-positive things. So that's what the lysozymes are doing. And mast cells are antibiotics on surface that are involved in inflammation, allergic reactions. If you become an allergist, then mast cells will become half of your life dealing with those. Okay, finally we have the bicarbonate buffer, and the bicarbonate buffer is something that's very important to understand. And it kind of goes back to what I was explaining over here with the medulla oblongata stuff. But the bicarbonate buffer is this equation, and you just want to remember Le Chatelier's principle, which is that when you have too much carbon dioxide, right? When you have too much carbon dioxide, you're going to push this equation this way. And when you have too much H+, right, you're going to want to push this equation this way. Okay, so when I have too much H+, my pH goes down, right? My pH goes down. And then I increase my respiratory rate because I want to get rid of this carbon dioxide, right? So when I have too much H+, this equation goes this way. I have too much carbon dioxide, and I need to breathe faster to get rid of this carbon dioxide. And when I increase my pH, basically this H+ is going down. So I'm going to force my carbon dioxide to produce more protons. And now I don't want to breathe faster because I have fewer carbon dioxide molecules, and so I'm going to decrease respiration to make sure the carbon dioxide doesn't leave my body. If that explanation of Le Chatelier's principle doesn't work for you, just memorize this, because that's kind of what you need to know. I won't say that it's high yield, but you probably will get a question or two on this, so it's kind of good to know. Okay, next we have the cardiovascular system, and this is actually the very first unit that we had in med school, so I really like this topic. So I would say that it's a lot easier to understand the blood pathway if you're looking at a diagram. When I first looked at this, I was trying to understand from this, and it was very confusing. So I would say that it's best to look at a diagram and understand it. I think this should work. So okay, so the idea here is that there's these two veins that drop blood into the right atrium, right? There's the superior vena cava and the inferior vena cava. And that goes into the right atrium, and that sends blood. All the blood from your body goes through this into the right atrium, right? Now the right atrium sends blood through the tricuspid valve into the right ventricle, right? Just remember that this is all deoxygenated blood coming from your inferior and superior vena cava into your right atrium, then your right ventricle through the tricuspid valve. Now it's in your right ventricle. The right ventricle is going to send it through the pulmonary artery, right? And it's going to go left and right, through your pulmonary artery to your lungs, right? So it's going to go to your lungs. This is your lungs right here. And so this is all the deoxygenated blood is going through your pulmonary arteries to the lungs. And then it becomes oxygenated, and it comes through the pulmonary vein back to the left atrium. Okay, so now the newly oxygenated blood from your lungs are going from your pulmonary artery, are going through your pulmonary vein to send oxygenated blood to your left atrium. Now your oxygenated blood from the left atrium is going through the mitral valve to your left ventricle. And the astute observer will notice that this left ventricle has a really thick wall, right? A thick myocardium. And the reason for that is because this left ventricle is sending blood, the oxygenated blood, to the aorta, right? Throughout the entire body. And so this has to be a really thick wall because this ventricle is responsible for sending blood to the entire body. So it's going to be really, really harsh push through the aortic valve into the aorta, which the aorta is the very first artery after the heart, and then sends blood to the rest of the body through a variety of arteries that then go into arterioles, that then go into capillaries, that then go into veins, and then finally go to the heart, right? Let me repeat that. So it goes through arteries, then arterioles, then capillaries, then venules, and then veins, and then back to the heart through the superior and inferior vena cava. All right. Hopefully that makes sense. So this is basically, I just covered all of this. And if you understand that, then that's a huge, huge important thing in the cardiovascular system. Now you should know the electric conduction. I can show you an image for that too. Okay, so the electric conduction happens from the SA node. So happens from the SA node over here, and then goes to the AV node, right? And then, I don't know if this is the best. I think it might be easier to just read this. So it goes from the SA node, which is the pacemaker of your heart, to the AV node, and the AV node is kind of slow. So it's like, it'll wait for the electrical signals that's going from the SA node to the AV node and kind of slows it down, and then goes to the bundle of His and then the Purkinje fibers, right? So you can either remember this or you can remember the stab a big pickle mnemonic. I kind of just remember this. And you should know that the SA node is the pacemaker of the heart. Now, what if the SA node stops working, right? Because your heart is just responsible for beating all the time, no breaks, because if it takes a break, you die, right? So if this happens, then there's backup pacemakers, and that's the AV node, the bundle of His, the Purkinje fibers. And so these are responsible for creating electrical signals that constantly tell your myocardium to keep beating your heart. Okay, next we have blood pressure. Blood pressure can be systolic or diastolic. And systolic is when you're contracting your heart, and so there's higher blood pressure because you're contracting your heart, trying to push blood outside of your heart. And diastolic is when you're relaxing your ventricles, and so the blood pressure is going to be lower. Should know that the AV valve closes during systole, and the SV valve closes during diastole, but it should make sense if you understand the blood pathway. Okay. Next, we have the normal blood pressure. And normal blood pressure is usually 120/80. When you increase the blood pressure, you increase this chemical called ANP, and that's responsible for decreasing the blood pressure. When you decrease the blood pressure, you increase this chemical called aldosterone, or an ADH, which we already talked about. They're hormones, and that's kind of responsible for bringing the blood pressure back up. Next, if you increase the osmolarity, like remember I said, you increase the ADH, which sends out a lot of those solutes through the urine. Cardiac output is how much blood is being sent outside by your heart per minute, and it's equal to the heart rate, which is how many beats per minute, times the stroke volume, which is how much blood are you sending out, right? And that gives you your cardiac output, which is how much blood are you sending out per minute. And the SV, the stroke volume, is how much blood you're sending out per beat. If you multiply the two, you get cardiac output. Okay, let's talk about the vasculature real quick, which I kind of talked about already. Arteries are these thick, muscular, elastic blood vessels that are responsible for recoil, that allow recoil and help propel the blood forward. Why are they thick and muscular? Well, because they're sending blood through the rest of the body, and so they have to be like very, very thick. And so if you look at any pathology slide, you can kind of tell the difference between arteries and veins, because arteries just have these thick walls. Arterioles are smaller muscular arteries. This is when they start to separate into smaller blood vessels. And then we get really, really tiny blood vessels, which are capillaries. And they're so tiny that they're actually just only one cell thick. And the reason why they want this is because you want to kind of slow the blood flow down, and you want the capillaries near these tissues so that there can be exchange of gases. The oxygenated blood is giving oxygen from the hemoglobin to the tissue, and the tissue is giving its carbon dioxide, that's a result of cellular respiration, to the hemoglobin so that it can go back to the lungs through the heart. Next, we have veins, which are thin walls and inelastic. They can stretch to accommodate blood, but they don't have recoil, so they're not like going back and squeezing the blood through. They're not helping to pump as much. There is surrounding muscles that do help, but not as much as the arteries, which are really, really trying to propel the blood forward. And then there are venules, which are very small veins. And the venules will go back to the superior and inferior vena cava. Okay. Actually, sorry, I don't know why this is in reverse, but it should be capillaries to... Yeah, so it should be from capillaries, you go to venules, and then you go to veins. So I guess if I was going to write this, it would be heart goes to artery, goes to arterial, goes to capillary, goes to venules, and then goes to veins. So veins, and this is the superior and inferior vena cava that then goes back to the heart. I hope that makes sense. Okay. Okay, next is blood. Blood is considered a connective tissue. It's made of erythrocytes, which are red blood cells. It's very important to know that all the cells in your body have a nucleus and have a nucleus except for the red blood cells. Okay, they don't have a nucleus, they don't have mitochondria, they don't have organelles. They're just kind of responsible for containing hemoglobin, and hemoglobin is the important protein for carrying oxygen. Erythrocytes, red blood cells, are formed in the blood, in the bone marrow. Next, we have hematocrit. Hematocrit is the percentage of blood that's composed of red blood cells. So it might be like 55% or something. You should know that not all the blood is red blood cells. There's also that liquid that the red blood cells is within. Now we have leukocytes, which are white blood cells, and this is very, very important in the immune system. And we'll get into this a little bit more later. But leukocytes include granulocytes, which are neutrophils, eosinophils, basophils. We have agranulocytes, which are lymphocytes. Lymphocytes are like B and T cells that provide specific immunity. So when you have a virus, it makes a specific antibody or specific reaction to that virus. Whereas neutrophils, eosinophils, and basophils, they respond the same to all different types of inflammatory reactions or viruses or bacteria. Monocytes and macrophages are kind of responsible for digesting the foreign matter. So you can think of macrophages as kind of eating something, eating bacteria. And it's important to remember that neutrophils, eosinophils, and basophils provide non-specific immunity. Okay, next we have thrombocytes, or platelets. Thrombocytes are important for cell fragments and coagulation. They're very, very important for coagulation. Coagulation is kind of when you create a blood clot. And so let's say you have some sort of bleeding in your endothelium, right? You have some sort of crack, and so your blood starts to leak out of that blood vessel. You want platelets to come and plug up that hole, and that's called coagulation. Now there can be a problem with this, which is that if you have too much coagulation, you could create a blood clot, and that blood clot could embolize, and that could cause a stroke if that goes to the brain, for example, right? And so sometimes when somebody is having too many platelets respond to these types of events, you give them anticoagulation medication to make sure that they don't have this sort of stroke or embolism or thrombosis event. You don't need to know too much about that though. Okay, for the purpose of the MCAT, by the way. You'll have to know that when you go to med school. Okay, blood type, antigens. Antigens are surface proteins on red blood cells. It's very important to know this because when you have a transfusion, right, when you're giving somebody your blood, you don't want to have like kind of you don't want to be giving red blood cells, which will have a bad reaction in their body because they'll create immune cells against your red blood cells. So you want to look at the antigens that are produced, the antibodies that are produced, and who you're able to donate to. So, and Rh factor is also something you want to consider. So if you are, for example, blood type AB, that means that the antigens that are being produced against your red blood cells are AB, and both A and B against your red blood cell surface, and you're not going to be producing any antibodies. And so you can only donate to other people who have AB because if you give to anybody else, they're going to have antibodies against either the A or the B, right? But you can receive from anybody because you don't have antibodies against anything. You're okay with receiving anything because you have both A and B antigens. And so your immune system thinks that A and B are your own self antigens. And so it's not going to attack A or B or AB or O. But let's say you're O. That means that you don't have any antigens on your red blood cells, but you have anti-A and you have anti-B. So you have these immune cells, these B cells that are producing antibodies that if it sees A, then it's going to attack it. If it sees B, then it's going to attack it. So you can't receive from anybody besides O, right? Because if you receive from somebody with blood type A or blood type B, you're going to produce you're going to produce antibodies against that. But you can receive only from O, but you can donate to anybody because you don't have any antigens on your red blood cells, and so nobody is going to produce any sort of immune reaction against you. That's how you think of this. This took me too long to understand. Next, you have coagulation. Coagulation is when the endothelial lining of the blood vessel is damaged. So this is what I was talking about, coagulation, by the way. I introduced it a little too early. And so when your endothelial lining of blood vessel is damaged, collagen and tissue factor is going to be exposed. And this is how you produce fibrin and thrombin, right? And fibrin and thrombin are responsible for kind of clotting up that place so that you don't have loss of these blood cells. And these clots are broken down by plasmin, okay? So eventually you're going to have to break down this clot once the endothelial lining has healed. And how do you break them down? With plasmin. Okay. Finally, we have fluid balance. We're almost done, but fluid balance is hydrostatic pressure and osmotic pressure. Osmotic or oncotic pressure is when you have these solids that kind of suck in pressure, and hydrostatic pressure is the pressure created by the fluid. And so if you think about kind of like this bucket where you have a lot of solids, I'm sure you've seen something like this, right? And like two solids here. The water, if there's like equal amount of water on this side and this side, the water is going to want to come to this side, right? From the site with fewer solutes to the site with more solutes, because these solutes kind of have this oncotic or osmotic pressure. Hydrostatic pressure, let's say like I did the same thing, but there's way more fluid on one side than the other, right? Then there's going to be hydrostatic pressure, which is going to send the water from more to less, and that's kind of the idea. Okay, oxygen is carried by hemoglobin, and carbon dioxide mostly exists in the bloodstream as bicarbonate, or HCO3. And we have this bicarbonate buffer, and that's kind of something that I've already went through, so just remember that. Okay, next we have the immune system, which is pretty exciting stuff. This is very high yield and a lot of information. So yeah. Make sure you understand everything so far. Just pause the video if you need to. Yeah. Okay, so we're talking about the immune system, innate immunity, and adaptive immunity. So innate immunity is defenses that are always active, but not specific. So by not specific, it means that the specific antigen that's invading your body, the innate immune system is going to have the same response to all of that. So example is on your skin, your mucus, your stomach acid, your tears, all of those are examples of innate immunity. Adaptive immunity is when defenses take time to activate and are specific to the invader, so specific to the species of bacteria, for example. Okay, let's talk about the innate immune system. So we have non-cellular and cellular immune defenses, and the cellular is actually very high yield, but non-cellular innate immune system includes skin, mucus, lysozymes, complement system, and interferons. So the skin is the physical barrier, and a lot of people don't think about the skin as a actual part of your immune system, but if you think about it, your skin is preventing a lot of bacteria from invading. And so there's a physical barrier, and there's also antimicrobial enzymes like defensin. The mucus, if you remember the mucoescalatory elevator that we talked about, mucus enzymes kind of trap pathogens, and in your respiratory system, so in your lungs, the mucus is being propelled upward by cilia via this mucociliary escalator. Lysozymes are in your tears and your saliva, and this contains some sort of antimicrobial compound. So if you think of something getting into your eyes, for example, like some dirt or something, you have these tears, and that contains antimicrobial compounds. Same thing when you're eating something and you're salivating, that saliva is helping to protect your body. Complement system is essentially this very cool system. It's kind of like this complex, complement complex that punches holes in the cell wall of bacteria, and it makes them osmotically unstable, and that leads to their lysis. It also triggers this thing called opsonization, which is kind of like its tagging system that helps other immune cells destroy the bacteria. So complement system is this very powerful system that the innate immune system uses in order to destroy bacteria. Okay, the last thing is interferons, and when a cell is virally infected, this gives off interferons, and that interferes with the viral replication and dispersion that viruses need in order to invade the host organism. Next, we have cellular innate defenses. So cellular innate defenses include macrophages, MHC1 and 2, dendritic cells, natural killer cells, and granulocytes. Remember, I said that this is high yield, so definitely know this. Macrophages are things that ingest pathogens and present them on MHC2. They secrete cytokines. MHC1 is present in all nucleated cells. They display endogenous antigens to the cytotoxic T cells. So essentially, this is a very important concept, and it's essentially true that every single cell in your human body that has a nucleus, so not red blood cells, they have MHC1, right? MHC1 is like this receptor, and it displays the endogenous antigen, so the antigen from inside your own body, to the cytotoxic CD8 T cells. And these T cells will then kill off the cells that display an antigen that's not endogenous, right? So MHC1, again, is on all your nucleated cells, and they display endogenous antigens to your cytotoxic CD8 T cells. If these CD8 T cells notice that there's something wrong with these endogenous antigens, or that there's not enough of these MHC1 molecules, or there's something wrong with this, then this T cell will kill off that cell. MHC2 is only present in these antigen-presenting cells, so very specific cells like macrophages, dendritic cells, B cells, epithelial cells, right? There's very specific types of cells that display MHC2. And these MHC2 display exogenous antigens, so things like parts of bacteria or things like that, to CD4 cells. And definitely know this one is CD8 and this one is CD4, and this is all nucleated cells, and this is only professional antigen presenting cells. Dendritic cells are antigen presenting cells in the skin. So remember, we talked about how MHC2 is on dendritic cells, so this is dendritic cells are on the skin. Natural killer cells attack cells low on MHC, including virally infected cells and cancer cells. So something that happens sometimes is that a virally infected cell might have fewer MHC1. So the natural killer cell will come and notice that there's something wrong, that there's fewer MHC1, and will notice that that's true, and will assume that that's a virally infected cell and kill your own cell. Sometimes that's true with cancer cells as well, so natural killer cells will kill those cells as well. Granulocytes include neutrophils, eosinophils, and basophils. These are called granulocytes because they have these granules, and they are activated by bacteria. Neutrophils are activated by bacteria, and they do this thing called phagocytosis, which is like eating the bacteria. Eosinophils are activated by parasites and allergens, and they increase the histamines. So they're very prominent. If you go to an allergy clinic, you'll notice that there's a lot of whenever you look at the pathology slides in the allergy patients, you'll see a lot of eosinophilia. And then there's basophils, and basophils are activated by allergens as well, and they inhibit blood clotting. Okay, the next thing is the lymphatic system. The lymphatic system, basically you have a lot of blood in your body. I think it's like 5 liters of blood, right? Let's just make sure that's numbers correct. How much blood in the body? There's 1.5 gallons or so, right? And so not all of that is in your circulatory system all the time. Or not all of that is in your blood vessels all the time. Some of that drains through the lymphatic system. And this is basically part of the circulatory system that has one-way vessels with these lymph nodes. And lymph nodes can be on your neck, your cheeks, right? Lymph nodes, and then they're important for these immune responses. So let me show you the image of the lymphatic system. What's a good image? Okay, so if we look here, we can see that, there's the whole blood system, and the excess blood that's going throughout your body, about 5% of that is getting drained through the lymphatic system. And the lymphatic system contains these little nodes, right? You can see these are nodes over here, and those are called lymph nodes. And if you've ever heard of a lymphoma, that's a cancer of the lymph node, and that results in like sometimes like swollen lymph nodes that you can feel on your neck. So the lymphatic system is really important for the immune responses, and it connects the cardiovascular system. It connects to the cardiovascular system via the thoracic duct in the posterior chest. So in the back of your chest, you have the thoracic duct, and it connects to the cardiovascular system. And it basically equalizes the fluid distribution, transports fats and fat-soluble compounds and chylomicrons. And sometimes when the lymphatic system is overwhelmed and it can't drain the excess fluid from your tissues, you'll have edema, which means that you'll have like fluid swelling in your body, and you'll basically see all this like your arm might be really big, or your legs might get really big because there's fluid buildup. Okay, the next thing is the adaptive immune system, and so we have humoral immunity, and humoral immunity is essentially antibody production by B cells, and it kills antigens when they're floating around in the fluid or the humor. And so this is the adaptive immune system, as in contrast with the innate immune system. So the adaptive immune system has B cells, antibodies. B cells make antibodies, right? B cells are made in the bone marrow, and they're activated in the spleen or the lymph nodes, and they express antibodies on their cell surface. Now, a special type of B cell is plasma cells, and plasma cells form antibodies. Plasma cells are activated B cells. So B cells are made in the bone marrow. They're in the spleen and lymph node, and they have antibodies on their cell surface. When you activate a B cell, you have a plasma cell, and plasma cells create antibodies. Now, an antibody will target an antigen, and an antibody contains 2 heavy chains and 2 light chains. And you'll have a constant region and a variable region, right? And so the tip of the variable region is the antigen binding region. And so this tip is specific to that antigen. Hypermutation can occur, and this is not super important for you to understand fully right now, but hypermutation is mutation of that antigen binding site on an antibody, and that results in varying affinities of the antibodies for specific microbes. So there's 5 different isotypes: IgM, IgD, IgG, IgE, IgA, and in different reactions you'll see a different response and different isotypes that are responding to that inflammation. But it's just important to know that hypermutation is kind of responsible for some of that variability in the antibody. Opsonization, again, remember, it's like this tagging system. It's when antibodies mark pathogens for destruction. Agglutination is when pathogens clump together and they form insoluble complexes. And that's caused by opsonizing pathogens. So things that mark pathogens for destruction, they'll also clump them together. And finally, we have memory B cells. And this is kind of why sometimes when you this is the way that like things like the COVID vaccine are based on, is that you have these B cells that remember the initial exposure, and then the second exposure to the pathogen, it doesn't have to go through this whole process of making new antibodies and responding to the pathogen. Now it just kind of remembers because they have these memory B cells, and so the secondary response is now really rapid and vigorous. And that's why, for example, the COVID vaccine, they first give you like this innocuous like strain of whatever, like maybe the cell wall, or sorry, the viral capsid or something, right? And they'll give it to you in a vaccine, and then your B cells, they respond to it, and now you have memory B cells. And then if you actually get the COVID virus, then your memory B cells will be able to respond to that in a much faster way and much more vigorous way than if you had not gotten the vaccine. Okay, cell-mediated immunity is on T cells, and cell-mediated immunity is response to cells once they've already been infected by the antigen. So once you have a cell and the antigen or the virus or the bacteria has invaded it, you want this cytotoxic immunity, which will basically kill that cell because that cell's been infected, right? And so, T lymphocytes are made in the bone marrow. They mature in the thymus. and they coordinate immune system and directly kill the infected cells. This is called cell-mediated immunity. You can have positive and negative selection. Positive selection means that you're maturing only the T cells that can respond to the presentation of antigen on MHC. So you're making sure that the T cells are only responding to the antigens, right, from the MHC. You don't want like these T cells to be killing off your self antigens, like the healthy cells, right? And so positive selection means that you're maturing only those T cells that are able to respond to the antigens that are like bad antigens, right? Negative selection means you cause apoptosis in the T cells that are self-reactive, right? So positive selection means you're promoting growth of good T cells, and negative selection means you're destroying T cells that are self-reactive. Next, we have helper T cells. These are CD4 T cells or Th. These are very important. They coordinate kind of like the rest of the immune system besides the cytotoxic immunity. And there can be Th1 or Th2. Th1 is responsible for creating this interferon gamma, so IFN gamma, and Th2 activate B cells in parasitic infections. Okay. So cytotoxic T cells are CD8 T cells. I've already told you about this. Suppressor T cells are these interesting set of T cells. So I've talked about there's helper T cells, cytotoxic T cells, and finally there's suppressor T cells. Suppressor T cells kind of do the opposite of what I've been talking about. They kind of downregulate the immune response after your infection is over. You don't want to be in this like hyper state of like killing things all the time. So suppressor T cells or T regulatory T cells, they kind of promote self-tolerance. And if they're defective, then you can get some autoimmune conditions. Memory T cells does something very similar to what memory B cells do. Autoimmune condition is when, self-antigen, like your own body, something in your body is recognized as foreign. And so the immune system attacks that. Allergic reactions is like a non-threatening exposure that results in an inflammatory response. So like an example is if you eat peanuts and your body thinks that that's like a antigen, that's a really harmful antigen, whereas it might not be, then that's an allergic reaction. And immunization is what I talked about, is that you want to induce active immunity. Passive immunity is kind of when you're transferring the antibodies to the individual rather than letting the individual develop their own antibodies. These last way shorter in time period. So immunization might last, like, for example, a whole year, whereas passive immunity might just last a few days or something. Example of passive immunity is when a mother gives her child breast milk. Okay. Whew. Next is the digestive system. So, yeah, I actually really like this topic. So digestive system, we can have intracellular or extracellular digestion. Intracellular is like oxidation of glucose and fatty acids through cellular respiration to make energy. And extracellular is just the process in which nutrients are absorbed from your food. Mechanical digestion is different from chemical digestion. Chemical digestion is like you're cleaving bonds within a peptide or protein or glycogen. Mechanical digestion is when you're breaking physically your large food molecules into smaller food molecules. So when you're like crunching a piece of broccoli, that's mechanical digestion. Peristalsis is, the rhythmic contractions of your gut tube. So if you actually like listen to somebody's stomach, and like if you tap and listen, you can actually hear the rhythms of the gut tube. And so this is increased peristalsis by the parasympathetic nervous system and decreased by the sympathetic nervous system. Remember, parasympathetic nervous system is all about rest and digest, and peristalsis is kind of the process of digesting, right? There's these rhythm contractions as you're digesting the food through the gut tube, and so that's why peristalsis is increased by the PNS. Okay, the digestive pathway is a lot easier to understand, I think, than the heart pathway, the blood pathway. You have your oral cavity or your mouth. It goes through the pharynx. Pharynx is where you have both food and air. Then it goes into your esophagus. Then it goes into your stomach. Your stomach is going to release a lot of HCl, a lot of acid, and it's going to be really, really acidic so that you can break down that food. It's going to go to the small intestine, then the large intestine, then the rectum. Okay, you have your oral cavity, and oral cavity is where you have mechanical digestion. Like I said, when you're chopping down on a broccoli, you have amylase and lipase, which kind of starts the process of digestion of food. And you might be like, how do we have those enzymes in our mouth? It's actually from the saliva that you're salivating when you're eating something. The saliva has amylase and lipase that starts to break down some of the fats. Food is formed into a bolus and then is swallowed. That's just what we call the food at that point. Pharynx connects the mouth to the esophagus, and epiglottis prevents food from entering the larynx. So if there's an issue with your epiglottis, you might have food in your larynx, and that's a really bad problem. You might like aspirate some food, and that can cause, that's like when you have a baby who's aspirating a peanut, for example, and then, that's a really big problem. The esophagus is something that, is like this pipe, and it propels food to the stomach using peristalsis. The top third has skeletal muscle and is under somatic control, and you should remember what somatic control is from earlier in this video. And the bottom third is smooth muscle, and the middle third is a combo of both. The middle and the bottom are under autonomic control. So if you think about it, next time you're eating, you might think about like, oh, am I in control of the digestion of my food, and at what point am I not digesting? And that can be a cool experiment for you. Okay, like I said, the stomach has a very acidic environment. It has 4 parts: the fundus, body, antrum, and pylorus. The fundus, body, antrum, pylorus. The enzyme pepsin is very responsible for breaking down protein. So I remember this by P and P. Secretory cells that line the stomach include mucus cells, chief cells, parietal cells, and G cells. So mucus cells are something that produces mucus, right? And mucus protects the stomach wall from the acid. So you don't want the stomach to actually get damaged from all the acid that's producing. So that's why you need mucus. And mucus has a lot of bicarbonate, which is, as you'll remember, is basic, and so it'll protect the stomach wall. The next thing is chief cells. Chief cells release pepsinogen, which then is formed, is converted to pepsin, which breaks down proteins. Parietal cells are very, very important, and they release HCl, so they actually release the acid, and they also release intrinsic factor. And so if you don't have parietal cells, this goes beyond what you need to know, then you might not produce enough intrinsic factor, and you'll get less vitamin B12 absorption, and you'll get this thing called anemia of chronic disease. And finally, we get G cells. G cells secrete gastrin, and that's a peptide hormone that increases the HCl secretion and increases gastric motility. So it'll help you kind of move the food down. And G cells basically tell parietal cells to secrete more HCl. Okay. Now, food particles that have been processed in the stomach are now called chyme. So if you remember from the mouth, it's called bolus, and now it's called chyme. Chyme exits through the pyloric sphincter, which is like kind of the last part of the stomach, and is it's a sphincter, and it goes into the duodenum, the duodenum, and the duodenum is the first part of the small intestine. Okay. Next we have these hormones. It's kind of important to know this. So ADH and aldosterone is going to increase your thirst, because basically it's sensing that there's less volume of fluid in your extracellular space, and so it's going to tell yourself like you need to drink a little bit more water. Next is glucagon and ghrelin. Glucagon and ghrelin both tell your body that you're hungry and you need to eat some food, and leptin and cholecystokinin increases satiety. The duodenum is the first part of the small intestine. It's very basic. It kind of needs to be, because that acid might come from the stomach, and so you don't want the duodenum to be I guess like damaged from that acid, right? So so you have disaccharides, you have aminopeptidase and dipeptidase, and you have enteropeptidase. Unfortunately, these are not too difficult to understand. Disaccharadases, they are brush border enzymes that break down all these disaccharides into monosaccharides. Aminopeptidases and dipeptidases, they're proteases, basically peptidases, and enteropeptidases activate the trypsinogen and procarboxypeptidase. I'd say that like kind of the important thing to understand is that there's enzymes in the duodenum that helps chemically break down your food. Now, the hormones in the duodenum are secretin and cholecystokinin. Secretin is a peptide hormone, and it stimulates the release of pancreatic juice. And that pancreatic juice is basically helping you to break down your food. This slows motility a little bit. Cholecystokinin stimulates bile release from the gallbladder. And that kind of helps again, and cholecystokinin also helps you release pancreatic juices, and also, if you remember, increases satiety. Okay, the next thing we have is absorption and defecation. So the jejunum and the ileum are part of the small intestine and are primarily involved with absorption. And the small intestine is lined with these things called villi. And villi contain microvilli, and that kind of helps you digest some of the things. So villi, the capillary bed helps you absorb water-soluble nutrients, and the lacteal helps you absorb fat-soluble things, and it sends it to the lymphatic system. So you can remember that fat-soluble are ADEK, and water-soluble are all other vitamins. Yeah, and water-soluble, because they're water-soluble, they can enter the plasma directly. The large intestine absorbs water and salts and form feces. The cecum is basically, I think it might be helpful if I show you digestive tract. Okay, it might be helpful if I show you this. So, all right, so you have your tongue, your mouth, it goes through the pharynx, and the epiglottis, make sure it doesn't go through the larynx. Then you have the esophagus, right? And it goes through your thorax. And then through your diaphragm, your esophagus goes, and it goes into the stomach, right? This is kind of like the pyloric sphincter over here, right? And it goes into the small intestine. The small intestine has the what is it? The duodenum, the jejunum, the ileum, and then you have your large intestine, which is like this thing, right? And the large intestine is the last part before you get to the rectum and the anus. Maybe we should do this one. Okay, so you can see that you have the appendix over here, you have the large intestine, and then you have the rectum and then the anus, right? Your liver is over here, and it's going to have bile. It's going to create bile. That bile is going to be stored in the gallbladder and then released into the duodenum. Your pancreas is right over here, sort of deep to the stomach, right? And this pancreas is going to release pancreatic juice. And this duct over here this part that goes into the duodenum from the gallbladder and the pancreas, this part over here is called the ampulla of Vater. Okay. Whew. All right. Now we have the structure of the colon. So the colon is ascending, transverse, descending, right, and then sigmoid. And then you have gut bacteria. Gut bacteria is responsible for producing vitamin K and biotin. Finally, let's talk about the accessory organs that I mentioned. So you have the pancreas, and the pancreas produces pancreatic juices that contains bicarbonate, amylase, peptidases, lipases. So it's just all these things that help break down your food, and bicarbonate helps protect your duodenum from the acidic stomach. The liver produces bile, albumin, clotting factors, right? Processes nutrients. So if you drink a lot of alcohol, somebody might tell you like, oh, you're making your liver do a lot of work, because it's the thing that's kind of detoxing alcohol, drugs. Liver receives blood from the abdominal portion of your digestive tract via the hepatic portal vein. And then we have the gallbladder, and the gallbladder stores and concentrates bile. So it's storing this bile that's produced by the liver, and the CCK, right, cholecystokinin, stimulates bile release into the biliary tree, which merges with the pancreatic duct. And if you remember, when these two merge with the pancreatic duct and the biliary tree, that goes into the duodenum via the ampulla of Vater. Okay? And hopefully you remember that this is endoderm. These are endoderm organs because they're endoderm. All right, making good progress. So we're almost done with this whole section. Next thing is kidney and skin. So let's see if I can find a good diagram for you. Okay. Sorry. This might be too complicated. Let's start with a simple one. So very simply, you should know that there's the kidney, and the kidney goes into the ureter. Then you have the bladder, right? The bladder over here, and then you have the urethra, which how you eject this urine, right? The kidney is producing this urine for the ureter. And I heard this really good quote from my teacher a couple months ago, which was that the kidney is not thinking about the kidney as producing urine is the wrong way to think about the kidney. The kidney is really responsible for maintaining your whole body's homeostasis and fluid and making sure like the amount of sodium and potassium and bicarbonate and everything in your body is right? So it's doing all this understanding of what your body needs and what it needs to excrete, and then makes the urine in order as a byproduct in order to maintain homeostasis. And so that's kind of a good way of thinking about what the kidney do. And so the kidney has this thing called the Bowman space. Let's see if I can show you here. So, okay. Bowman space kidney diagram. Okay, so you have the, let's look here. So you have the kidney, right? And the kidney has these little nephrons. Let's show you that instead. Okay, so then this is a nephron, which is kind of like the functional unit of a kidney. And it's kind of responsible for the whole process of absorption and of secretion and reabsorption. And so whatever goes through here is going to be released as urine, and there's going to be cells throughout that are going to be trying to reabsorb the nutrients that are going through here and also trying to secrete things, right? So we call this the tubular fluid, and then it's going to be released by the urine. And we're going to have cells all throughout that are right next to these that are trying to reabsorb the nutrients from the tubular fluid and put it back into the bloodstream. So I'm going to call this the bloodstream over here. So that's called reabsorption. And then there'll also be cells that are just secreting like ions and nutrients into the tubular fluid to be excreted via urine. So if you understand that, you'll know that first there's this glomerulus, right? Glomerulus is the first part, which kind of, this is the glomerulus, and this is all blood over here, right? And so the glomerulus, through this process, because there's, all this liquid and all these nutrients, so this is going to pass through the glomerulus, right? And this space in between the blood and the glomerulus is called the Bowman space. Okay, so it goes through the Bowman's capsule, right? So this thing over here, the blood, is called glomerulus, and this thing over here is called the Bowman's capsule. So my bad. So the blood is called the glomerulus, and the capsule is called Bowman's capsule, and the space between the blood and the Bowman's capsule is called the Bowman's space. And you should know that it goes from the glomerulus to the Bowman's capsule via, hydrostatic pressure and oncotic pressure and etc. So now that it enters here, it's tubular fluid, right? It's tubular fluid that'll eventually be released as urine. But we don't want this to go as urine as is. So there's going to be a process of reabsorption and secretion into and out of this tubular fluid. And so the first part is called the proximal convoluted tubule. Next we have the Henle's loop. Henle's loop is consisting of thin descending and thick ascending limbs of the Henle's loop. Next, we have the distal convoluted tubule, just like we had a proximal convoluted tubule here. Proximal means near and distal means far. And then we have the collecting duct, right? We have the medullary connecting duct and the, we have the cortical collecting duct and the medullary collecting duct. Okay, so that's kind of the nephron anatomy that you need to know. Sorry if that was not as well explained as I wanted to. We have the kidney. The kidney contains a cortex and medulla. And so this is kind of what I was talking about, is this part is the outside of the kidney, which is the cortex, and then we have the inner part, which is the medulla. And so the nephron goes from the cortex to the medulla and kind of back to the cortex and then back inside the medulla. Urine is collected in the bladder and excreted through the urethra. The nephron is the functional unit of the kidney, kind of like I said. The renal portal system is how blood flows through all the way to the glomeruli. So blood goes from the renal artery to the afferent arteriole to the glomeruli, and then it then goes to the efferent arteriole. So like all this filtration happens in the glomeruli through this whole thing, right? But this blood from the glomeruli go to the efferent arteriole, then the vasa recta, which surrounds the nephron, and then finally goes out the kidney through the renal vein. Filtration is that process in which the Bowman's capsule moves solute from the blood, and that's called filtration, right? Remember that the glomerulus contains all this blood. Here, I'll show you. So this is the glomerulus over here, this blood, and let me do it in red. So this is the glomerulus over here, right? And this is the Bowman's capsule over here. See over here is Bowman's capsule. And so you can see that, you know, the solutes in the blood over here need to go to the Bowman's capsule here, and that process is called filtration. Okay, secretion is when you have movement of solutes from the blood into the tubular fluid, and reabsorption is when you're going from the tubular fluid or the filtrate to the blood. From the filtrate to the blood is called reabsorption. The kidney is very responsible for regulating pH of the blood and kind of doing all these things with bicarbonate and H+. Aldosterone is synthesized in the adrenal cortex, and it kind of is responsible in this aspect, in this area, right? And aldosterone is responsible for making sure that potassium is secreted into the urine, and sodium and water are reabsorbed from the tubular fluid. That's what aldosterone does. And so what it's basically doing is increasing H2O reabsorption by increasing the sodium reabsorption. And if you understand that, then you understand that your aldosterone is basically encouraging your nephron to preserve water. And so there's no change in blood osmolarity because you're also reabsorbing sodium, but you're also increasing blood pressure because you're increasing the amount of water that's being reabsorbed. Finally, we have ADH, or vasopressin. And ADH is a peptide hormone that's synthesized by the hypothalamus again and released by posterior pituitary, and it increases the permeability of the collecting duct to H2O, which increases H2O reabsorption. So what you're going to get is very concentrated urine, and I kind of explained this already. Okay, now let's talk about the nephron a little bit more. So this is going to be talking about what parts of the nephron secrete and reabsorb what. So the proximal convoluted tubule is going to try to reabsorb everything that it can that's important. So you don't want to get rid of glucose in your urine. You don't want to get rid of amino acids. You don't want to get rid of vitamins, salt. Some water is going to be reabsorbed. So all of these things are going to be reabsorbed from the proximal convoluted tubule. Secretion, you'll have secretion of H+, K+, NH3, urea. The descending limb of the loop of Henle is permeable to H2O and not salt. And so basically, this tubular fluid, it's going to be going down and down. And what's happening is that the filtrate moves into more and more concentrated medulla, right? Because water is being reabsorbed. So water is being reabsorbed, so moved out of the descending limb. And so we're getting more and more concentrated tubular fluid down here, right, in the descending limb. And what's happening in the ascending limb is that it's permeable to salt and not water. So because we're coming up with this really, really concentrated thing, now the salt is going to be reabsorbed passively. So we're going to reabsorb the salt until basically the salt has been reabsorbed here. So it's not as concentrated when it goes back up into the distal convoluted tubule. So you might ask, like, oh, what was the whole point of that? Because basically we went down and we brought back water, and then we went up and we brought back salt. And so we're kind of, we didn't really change the osmolarity of that fluid that much. And the reason is because of this thing called countercurrent multiplier system, which is that that whole process creates maximal reabsorption of water. Whew. All right, next is the distal convoluted tubule, and that responds to aldosterone, and that creates salt reabsorption and waste product excretion. And finally, we have the collecting duct. And the collecting duct is responsive to both aldosterone and ADH. It has variable permeability, and it basically makes sure that the right amount of H2O is being excreted based on the body's needs. Okay, next we have the bladder, and the bladder has a couple of different has a muscle and then two sphincters that you should know about. So the bladder has this detrusor muscle, and it's under parasympathetic control, and it's this muscular lining of the bladder. And then we have the internal and external urethral sphincter. The internal one is smooth muscle, and the external one is skeletal muscle. The internal one is parasympathetic control, and when you really need to pee, it's voluntary control, and that's external urethral sphincter. Okay, so the next thing that we need to talk about is the skin, and this is not as high yield as some of the things that we've already talked about. So I'm just going to encourage you to memorize some of these concepts. The epidermis, the Langerhans cells, the melanin, right? The epidermis, you should know what these layers are. They don't come up too often in most of the practice exams, but it is somewhat good to know what these layers mean. So if you look at maybe skin, epidermis, layers, yeah, let's see. Okay, this is a pretty good diagram that we looked at in med school also when we were learning about the skin. And you can see the keratin here, the granules here, and you can kind of see that this is the stratum corneum, this is the stratum lucidum, this is the stratum granulosum, this is the stratum spinosum, and this is the stratum basale. And the important thing about the stratum corneum is that there are no nuclei, right? You can see over here there are nuclei, but here it's just a dead keratin layer, okay? And so you just kind of want to look at this and make sure you memorize some of these layers over here. The Langerhans cells are macrophages that are antigen-presenting cells in the skin, and melanin are produced by melanocytes. That's what gives you your skin color and protects your skin from DNA damage that's caused by ultraviolet radiation. Caucasian people are actually the most susceptible to skin cancer because they have the least of this melanin. The same thing, you should probably know what the dermis layers are, and it's a lot easier if you kind of search up dermis layers. I wish that I did this when I was studying for the MCAT, but looking up images can really help you. So you can see that there's the papillary layer and the reticular layer, and there's the subcutaneous fatty tissue below that, right? And so you can see that there's nerves and capillaries over here, and so just make sure you memorize this a little bit. It's not too difficult, but it'll help you a lot. And finally, we have thermoregulation. Oh, sorry, hypodermis is what I was talking about, which is this subcutaneous fatty tissue. So you can see that fat and connective tissue, it connects skin to the body. And then we have thermoregulation. So you should know that, sweating, when you sweat, your sweat evaporates, that cools your body down. You have piloerection, which helps you warm up. Shivering is also something that you're like shaking in order to warm up. Then you have vasodilation. Vasodilation helps you stay cool, and vasoconstriction helps you stay warm. Okay. So now we're on to the muscular system. So this shows up a little bit more often than the skin. And first, we have to talk about skeletal muscle. Skeletal muscle is important for all these different things. It's under somatic control, so you can control it. It's voluntary. It's multinucleated. There's support and movement, blood propulsion, thermoregulation, striated, right? It's important to know that this is striated. There's red fibers and white fibers, and you should kind of know what the difference are. Red fibers are slow twitch, and white fibers are fast twitch. Right? This is oxidative phosphorylation, and this is anaerobic metabolism. Next, we have smooth muscles, and smooth muscles are respiratory, reproductive, cardiovascular, and digestive. And hopefully you know that this is involuntary, right? You don't control some of these things, right? You don't control your heart beating. You don't control like your stomach releasing acid. So these are autonomic control. They're uninucleated. And you should know that they can have myogenic activity even without any neural input. So your brain doesn't even have to be involved for some of these things. Okay. Next is cardiac muscle. Cardiac muscle is the contractile tissue of the heart, right? It's under involuntary control. You're not controlling your heart. It's uninucleated, but sometimes it's binucleated. And this is the muscle of your heart. It can display myogenic activity. And the cells are connected with these things called intercalculated discs, which contain gap junctions. This is helpful for moving across that electrical signal. And now we're going to talk about the skeletal system overall. Skeletal system, remember, is derived from the mesoderm, which is the movoderm. You have the axial skeleton, and you have the appendicular skeleton. And the axial skeleton is skull, vertebral column, rib cage, hyaloid bone, and the appendicular skeleton is bones of the limb, pectoral girdle, pelvis. You can have compact bone, which is very strong and dense. You can have spongy bone, which is lattice-like. You can have the bone marrow. The bone marrow is, remember, filled with these hematopoietic stem cells, and it's responsible for producing a lot of the red blood cells. The yellow part of the bone marrow is fat. You can have these long bones. It's not super high yield to understand this, but make sure you understand some of these things. Another thing that's kind of high yield is ligaments and tendons. Ligaments attach bone and bone. Tendons attach bone to muscles. Let's see, I think this is very high yield, which is that bone remodeling. So osteoblasts build bone. So I just remember osteoblast build bone. And then osteoclasts resorb bone. And resorption here means that it's breaking down the bone to produce calcium. And so parathyroid hormone is something that's responsible for increasing resorption of bone because there's too little calcium in the blood, and so it's going to increase the blood calcium by resorbing this bone. Vitamin D also increases the resorption of the bone and also increases blood calcium. And then calcitonin, if you remember, does the opposite. It increases bone formation and decreases the calcium in your blood. These are all kind of ways to regulate how much calcium is in your blood. Next we have cartilage, and that's kind of this firm and elastic tissue. Next we have joints. Joints can be immovable, movable, and there's synovial fluid, which kind of lubricates these joints. Let's look at more. Okay, now I want to talk about sarcomeres, which is kind of like the very high yield concept here. Sarcomeres are these basic contractile units that have striated muscle. They're called striated because if you look at them under a microscope, you can see like these lines, right? Unlike smooth muscle. And so you have thick myosin and thin actin filaments. Okay, so the thick myosin and the thin actin filaments are responsible for this contractile unit. And on the thin filament, you have, so on this thin actin filament, you have troponin and you have tropomyosin. Okay, that's found on the thin filament, and it regulates these interactions. So I can show you the diagram here. And so you have the myosin, right? You have the actin filament, which is this blue thing over here. And you have diagram. Troponin and tropomyosin diagram. Okay. So you can see that, you know, there's this actin, there's the tropomyosin, there's the troponin, and okay, actin, troponin, tropomyosin, myosin, calcium. So basically what's going to happen is, let's see. Okay, I'll get into the contraction and relaxation later, but just know that there's troponin and tropomyosin on this thin filament and regulates this actin-myosin interaction. We should talk about what the parts are here. So there's the Z line, there's the M line, there's the I band, there's the H zone, and then there's the A band. Just know what each of these mean. Z line is the boundary, M line is the middle, I band is only the actin filaments, and H band is only the myosin filaments, right? This is the I band over here, this is the H band over here. Next, this is the I band over here, this is the H band over here. And A band is the part that contains both, right? Okay, sarcomeres attach end to end and become myofibrils, and each myocyte contains many myofibrils. So you can imagine that, you know, there's another sarcomere here, and because they're attaching end to end, they're forming myofibrils, and each myocyte can contain many myofibrils. Next, we have the sarcoplasmic reticulum, and this is kind of a similar version to the endoplasmic reticulum, except calcium plays a really big role in the sarcoplasmic reticulum. Sarcolemma is the cell membrane of the myocyte, and T-tubules connect to the sarcolemma, and they carry these signals. Okay, and we're going to kind of understand a little bit more about this with this part. So contraction and relaxation begins at the neuromuscular junction, and it's where there's efferent neuron release of acetylcholine that binds to receptors on the sarcolemma, remember the cell membrane, and causes depolarization. And so depolarization then goes down to the T tubules and triggers release of calcium. Calcium binds the troponin, causing a shift of the tropomyosin and exposure of the myosin binding site on the actin filament. And then there's shortening of the sarcomere, and then this is called the sliding filament model. So when your sarcomere shortens, then you're basically you're basically sliding this filament. Let me try to find a diagram that might help you. Acetylcholine in muscle contraction diagram. Okay, this might be good. All right, so you can see that over here, you can see that there is this acetylcholine that's entering, right? This acetylcholine goes down through the T tubule, and it's getting released into the sarcoplasmic reticulum, right? It goes and binds over here, and it binds to troponin on actin filaments. And now the tropomyosin is going to physically move aside, right? Because the calcium has bound to the troponin, so the tropomyosin is going to stop blocking this. And so now we're going to be able to cause a contraction. Okay, I'd encourage you to kind of read through this diagram and make sure you understand everything, and also kind of just read through this and make sure you understand everything. But I highly encourage watching kind of a YouTube video to understand this, because when I first learned it, it was really complicated to me. Okay, almost done, last page. So this is genetics and evolution, and I really like this topic. So the first thing is alleles. Alleles are different types of forms of genes, right? So the gene might be eye color, but the allele might be brown or blue or green or gray, right? And if you have a dominant allele, then you only need one copy for it to be expressed, and recessive require two. Genotypes, there's the combination of alleles that you have at a given locus. So homozygous genotype means that you have two of the same, and heterozygous means you have two different. Phenotype is when you have an observable manifestation of a genotype. So the genotype might be big A, little a, and the phenotype might mean green eyes or something, right? Dominance can be complete or incomplete or codominant. So complete means that there's only one dominant allele. Codominant is that there's more than one dominant allele, and incomplete means there's no dominant allele, right? So you might be able to express both alleles. You might be only able to express one allele, or you might not be able to express either allele completely. And so that's what dominance means. Penetrance is the proportion of individuals who are carrying a specific allele that also express the associated phenotype. So sometimes people who have a particular allele might not actually express the phenotype that's associated with that genotype, right? And so the percentage of people who actually do express that phenotype when they do have that genotype is called penetrance. And expressivity is the variable phenotypic outcome of a genotype. So say somebody has the genotype for like extra fingers on their hand. They can either have like 9 fingers on their hand, or they can have 6 fingers on their hand, and that's expressivity. Genetic leakage is the flow of genes between species via hybrid offspring. Genetic drift is when the composition of a gene pool changes as a result of chance. So I'm sure you've done like those experiments of when you have like 10 people in a population, and you see like how do the genotypes differ, and eventually genetic drift will happen, right? And so you'll get one allele, one gene that just dominates. And that's called genetic drift. Founder effect is when, for example, you have like a bird that goes to a new island, and its genes are like kind of the thing that is responsible for creating a lot of the new genes. There's a lot of inbreeding as a result of founder effect. And then you should know that this is the taxonomic rank. It's not very high yield to know this, but it's still not that difficult to memorize if you remember King Philip came over from Great Spain. Okay, next we have Mendel's laws. There's the law of segregation and law of independent assortment. The law of segregation says that the two alleles for a gene segregates during anaphase 1. And the law of independent assortment means that these alleles that are separating don't have any impact on one another. So if you have an allele for little a, that doesn't affect the allele for little b on a different chromosome. This is not true for linked genes. If you have little a and then little b on the same gene, then that's going to have increased likelihood of being together in the offspring, right? But if you're on different chromosomes, it doesn't have any impact on each other. Okay, the next thing is we have experiments that have shown that DNA is the genetic material. Sometimes this shows up because in a passage they'll explain to you an experiment that's very similar to one of these experiments, and they'll ask for the outcome. And if you know these experiments, you'll kind of know what the outcome to expect is. The Griffith one showed that there's transformation was possible. So these bacteria were like basically picking up DNA from their environment, from the virulent strain in their bacteria, and were basically transforming their DNA. It's also, if you search up on YouTube these experiments, you'll find like 2 or 3 minute explanations, and those are really, really helpful. Avery, McLeod, McCarthy basically showed that the degradation of DNA led to a cessation of bacterial transformation, but when you degraded proteins, it did not. So it showed that DNA was the thing that was being transformed, not the proteins. And lastly, we have Hershey-Chase, which showed that when you radiolabeled the DNA, that was the only thing that was being found in the bacteriophage infected bacteria. So you inputted a lot of bacteriophages, and you saw that there was only radiolabeled DNA in that bacteria. Okay, this is different types of evolution, so you should know this: divergent, parallel, and convergent. And then we have nucleotide mutations. This is all very, very, very high yield for you to know. Nucleotide mutations can be point mutation, frame shift mutation, and the results can be silent, which means that there's a mutation, but it doesn't have any effect on the protein, on translation of that protein. There's missense and nonsense. Nonsense is when you have a stop codon, and so your amino acid chain might stop too early, and you don't actually translate everything that you need to, right? There's also missense, which means that one amino acid has replaced another amino acid. So you might need like methionine over here, but you get, I don't know, glutamine or something, right? Insertion or deletion can result in this thing called frame shift mutation, right? And so that's like moving the 3-letter reading frame. Sorry. So frame shift mutation is when you have moving the 3-letter reading frame, and you can get an insertion or deletion into the amino acid. So it's important to know, and a mistake that I just made is that this insertion or deletion is referring to the amino acid chain, and this frame shift mutation is talking about the mRNA that's producing that amino acid chain. So if you have, like, for example, A, T, C, right, and you add like an A over here, then that's an insertion of a nucleotide, and that's going to cause this to be A, T, and then C is going to be on the next one, right? And so this is going to be a frame shift mutation, and that's very dangerous because basically everything in the DNA or the RNA molecule is going to be changed because you're now moving that 3-letter reading frame. And so these are the results in the protein, and so you can get silent, which is no effect on the protein. You can get missense or nonsense, and then finally you can get insertion or deletion, which is the insertion of an amino acid into the amino acid chain. Okay, I hope that makes sense. Just make sure you review that so that it makes sense. Next, we have chromosomal mutations, and this can be like huge segments of DNA on a chromosome that are being affected. You can have deletion, duplication, inversion, insertion, or translocation. And I probably would encourage you to just look at an image. I can show you that now, because these are not too confusing to understand, but it was very confusing for me when I was first looking at it. So if you look here, these are very easy diagrams to kind of understand what's going on. This is a deletion over here. This is a translocation over here. This is a duplication over here. And this is an inversion over here. And if you understand these concepts, then it's very easy to kind of know what's going on. Okay, now we have analytical techniques. The Hardy-Weinberg principle is P plus Q equals 1, and P and Q are the 2 different alleles and how frequent they are. Now sometimes there's more than 2 alleles. Maybe there's like 10 alleles. So this is true under a certain number of conditions. And if you have 2 alleles and your population is stagnant and it's not changing and things like that, there's a lot of different criteria that you can search up, then this equation should be true. Okay. Recombination frequency is also kind of this important concept, and recombination is the likelihood that 2 alleles are being separated during crossing over in meiosis. And so I never really understood this concept until I started med school. And basically what's going on in recombination frequency is when you have a single chromosome, right, you might have multiple genes on it. Sorry. You might have multiple genes on your chromosome, right? And usually you'd expect that on this chromosome that these genes would stick together in the daughter cell. But what might happen is, if you remember meiosis, you can get crossing over, right? And so in crossing over, these two alleles might swap, right? And so recombination frequency is how often does that swap happen, because you have crossing over, like crossing over here. And so this gene is no longer attached to this gene on the same chromosome. How often does that happen? Well, that's actually directly equivalent to the closeness of these two genes. If these two genes are very close together, it's very likely that the daughter cell will get these two alleles together, right? But if these two genes are very far apart, these two alleles on the same chromosome, then crossing over might happen somewhere in here, and so there's going to be recombination. And so these two alleles might not end up together on the same chromosome in the daughter cell. Okay, next we have evolution. And so evolution is when you can have natural selection. Natural selection is kind of the main method of is the actual mechanism for evolution. Yeah, I don't think it's that important to know a modern synthesis model. It's just this idea that mutation and recombination are mechanisms for variation. It's not just mutation, but you can also get the recombination, which is kind of like the biochemical way that you can create variation. You can also have inclusive fitness. Inclusive fitness is that if a population meets certain criteria, then the allele frequencies will remain constant. And that's only when there's a lack of evolution. So when you learned about like the bird beak and the finch's beaks, right? There was evolution going on because the environment was changing. And if that's not true, if there's no like change in your environment, then you'll have inclusive fitness, which means that the allele frequencies will remain the same. Punctuated equilibrium, again, not that important. It's when you have very, very slow processes of evolution, and then you have this burst of evolutionary activity. You can have these different types of natural selection, which you should just read. It's not too important to know for the purpose of MCAT. But yeah, just know what stabilizing, directional, disruptive, and adaptive are. And then, yeah, I guess the last thing that we should talk about is the molecular clock model, which is the degree of difference in the genome between two species that are related to the amount of time that the two species broke off from the common ancestor. So I once did a research project in which I was looking at like this SOX11 gene between mice and frogs based on how far apart they broke off from the common ancestor, right? And you can kind of figure out how different the genomes are based on two species, based on like when their last common ancestor was. And that's kind of the molecular clock model. And so the further away their common ancestor was, the further back, the more genetic differences they're going to be. But if they're like, they very recently had a common ancestor, then it's more likely that their genome looks pretty similar. That's what that is. Okay, that's everything you need to know for the biology section. I really hope that was helpful. Just make sure you go through everything and understand everything that we went through. And I really, really appreciate your time. Thanks so much for watching and giving me your attention. I'll see you in the next video. Bye, everybody.